Hi, it's time for another math easy solution. Today I'm going to go over another hashtag MES Science video, and I'll look at MES Science 4. And this will be on a review of COVID-19, quote, virus isolation paper, and it's going to be the main one. Yeah, that was cited many times earlier in 2020. And yeah, even though it's been 3 years since COVID started, I haven't made a video on it until now because I just wanted to get caught up in biology and so on, and a lot of virology and so on. Yeah, before I got comfortable into making this video. Anyways, let's jump right in. And note, as always, if you don't have time to watch this whole video, you can skip to the end for summary and conclusions, or you can play this video at a faster speed. Top secret life hack. Your brain gets used to faster speed, and here's a tutorial I made over here. And also you can download and read the notes, as well as read the notes on a Hive blockchain. The links will be all in the description below. And you can also watch a video in parts, and timestamps of all parts are in the description. And also a new thing I started doing was adding video sections in a playlist. So I'll separate this, let's say, long video into several smaller videos, and you can watch those, and the links will be in the description as well when available, and in this video it will be available. Anyways, let's continue further. So hashtag MES Science video. So for reference, here's a list of the MES Science videos that I've done so far, which include links to the corresponding Hive posts, so on the Hive blockchain, and YouTube sections playlist. So and here's MES Science tutorials. This is the link. This one is to the YouTube tutorial. The YouTube playlist. And then YouTube playlist short URL, you can just go to mes.fm/science-playlist. And then here's just a recap. So the first one I did was on a Powerball gyroscope. The second one was on Vortex Math Part 2, and I should be doing parts 2 and 3, hopefully in the future. And then Part 3 was a 48-hour-long video on overview of mainstream biology, which was basically me trying to catch up on mainstream biology, because it's not my background. Yeah, my background is mathematics and engineering, but then so I had to get caught up in biology, and also progress and update. This was before I did part 3 or MES Science 3, and this was on regarding COVID-19 and masks video. Yeah, which was the initial plan, just doing a video on that, but then I ended up just doing a overview of mainstream biology first. And now MES Science 4, this is the current video, is review of COVID-19 virus isolation paper, and there's the link to the playlist, which I will separate this video into many parts and upload those gradually on YouTube. Anyways, let's continue further. So MES Research Note. In this video, I go over a methodical examination of the main paper on COVID, quote, virus isolation. The reason for this methodical approach is that it helps me to learn and understand a paper while also presenting it to the public. So while it may be a long and tedious video, I believe it will better crystallize the mainstream science involved in the fear-mongering of a new, quote, deadly virus that requires us to, quote, to obey, quote, health authorities. And with that, buckle up, folks. Alright, so now let's take a look at the topics to cover first. And so the first one is going to be on a mainstream overview of COVID-19. And then number 2 will be look at ChatGPT on the main paper on COVID virus isolation. And then look at the paper, A Novel Coronavirus from Patients with Pneumonia in China 2019. Then go over the paper abstract and look at it, then basically summarize it. Then I'm going to go over a paper introduction, then do a summary on it. And again, doing a methodical approach, pretty much sentence by sentence. Yeah, and it's part study for me, but also part identifying exactly what the paper is doing and claiming. And then then paper abstract summary, paper introduction summary. Then go over a quick overview of nucleic acids and genomes. And then also go over the paper methods, detecting, quote, viruses, and look at a summary. Then isolating the, quote, virus, and then do a summary. Then look at the electron microscope preparation, and then look at the summary for that. Then sequencing the, quote, virus and summary. And then RNA assumed to be from a virus or a, quote, virus, summary. Paper results, we're going to look at patient results, summary, detection, and isolation of a new, quote, virus. Summary, and then genetic characterization of the, quote, virus, and then look at a summary, and then we're going to go over a paper discussion, and then summarize that, and then finally I'm going to go over an MES summary, and then MES conclusions. And yeah, these ones will be basically summarizing the key parts of the paper in regards to, quote, virus isolation, and then basically go over some conclusions that I have derived from it. Anyways, continuing further. Yeah, so now let's take a look at the mainstream overview of COVID-19. So let's first go over the mainstream overview of COVID-19 as summarized by the main, by the mainstream narrative, or the, actually no, this word is good, by the mainstream narrative machine, which is Wikipedia. Yes, if you want a mainstream narrative on anything, just go to Wikipedia and it will tell you that. Although ChatGPT is right up there too now. So Wikipedia right here. Yeah, basically, just starting over here. So coronavirus disease 2019, abbreviated COVID-19, is a contagious disease caused by a virus, the severe acute respiratory syndrome, coronavirus 2, or SARS-CoV-2, or CoV-2. And also a note here, this coronavirus disease, abbreviated COVID right here. So CoV is for the coronavirus, and then ID, I believe that's identified in 2019. Yes. All right, continuing further. So the first known case was identified in Wuhan, China in December 2019. The disease quickly spread worldwide, resulting in the COVID-19 pandemic. The symptoms of COVID-19 are variable, but often include fever, cough, headache, fatigue, breathing difficulties, loss of smell, and loss of taste. Symptoms may begin 1 to 14 days after exposure to the virus, and at least a third of people who are infected do not develop noticeable symptoms. So yes, basically a third have no symptoms. Of those who develop symptoms noticeable enough to be classified as patients, most 81% develop mild to moderate symptoms up to mild pneumonia, while 14% develop severe symptoms, dyspnea, hypoxia, or more than 50% lung involvement on imaging. And 5% develop critical symptoms, respiratory failure, shock, or multi-organ dysfunction. And older people are at a higher risk of developing severe symptoms, and some people continue to experience a range of effects called long COVID for months after recovery, and damage to organs has been observed. Multi-year studies are underway to further investigate the long-term effects of the disease. All right, so frequency here is 676,609,955 cases. Yeah, basically almost 700 million supposed cases, and this is as of March 12th when I was first writing this up. And then deaths as of March 12th, 2023, supposedly 6,881,955. So basically almost 7 million supposed deaths. Note that the world population is currently at about 8.021 billion. And here's a breakdown I made of the above figures. Alright, so here is the breakdown I made. I made an Excel file, and you can download it, and then you can play around with it. It has all the calculations in there. So what I did was, first I made a relative percentage, and this is relative to each subsequent section here. So for example, you have 100% of the population, and then you'll have 8% are cases, and this is derived from this 676 million cases as a percentage of this 8 billion. So that will be 8%. And then, so that means 92% of the world have aren't a COVID case. In other words, they didn't, quote, test positive, whatever that means. All right, so now we have the next section here. So now we'll have, going back up here, at least a third of people who are infected, or a case, do not develop noticeable symptoms. So then this is a third is of this. So 1/3 of this 8% have no symptoms. Then you have this one is the remaining is the symptoms, 2/3. And then going further, you'll have 81% are mild to moderate of the ones that show symptoms. Going back up here. So of those who develop symptoms, notable enough to be classified as patients, then most 81% develop mild to moderate, while 14 are severe, and then the 5% are critical, and so on. So we have this. So we have 81% of these, so this is what I mean by relative. Yes, 81% of people with symptoms are mild to moderate. Then you have 14% severe, 5% critical, and now the deaths part is this is 31% of these would be deaths, and I used this deaths figure as well as calculating the critical the population with critical cases there, and then use as a relative of that. So 31% of the people with critical cases died, and then 69% didn't die. And again, this is as of March 12th, 2023. Going further, I made an absolute percent cases, and this one is, this one's actually absolute percent from cases. So this table I made, so you have cases right here, and then, so we start off from here. We're basically, we're going to start off, make this 100%. So we're going to ignore this, start off at 100%. And then we're going to, instead of doing relative to each one, we're going to do this all absolutes from the case. So in other words, so if you have cases like this, you'll have 33% or a third no symptoms, 2/3 symptoms. And then you'll have, of the cases, 50%, 54 is mild, 9% severe, 3% critical. And then supposedly you have 1% deaths and then 2% non-deaths of these critical ones. So you'll have 1% of all cases are deaths, which is very interesting. Interesting statistic, highly questionable though. Anyways, now let's look at the absolute percentage. And this is based on starting off from 100% population. So you have 100% population, you have 8% cases, 92% non-cases, and then 3% no symptoms, 6% symptoms. Then you'll have 5% mild to moderate symptoms. And then you'll have 1% of this entire world is severe. 0.3 is critical. You have 0.2% non-deaths of these critical ones. But then you'll have 0.1% of the population has died, supposedly, from COVID. All right, and then going, now here's the absolute numbers. You have 8 billion population, 7 billion have no cases, 76 million have cases, 225 million have no symptoms, 451 million have symptoms, and these again have the cases, because these ones would be counted as the no symptoms there. So you have basically almost, you'll have 7.5 billion plus no symptoms at all. So now you'll have 365 mild to moderate symptoms, 365 million, and 63 severe symptoms, 22 million critical symptoms, and then 15.6 million non-deaths of the critical, and then 6.8 million deaths supposedly. That's very, very interesting. All right, I just wanted to go over that overview. So these are some big but highly questionable numbers that need further investigation. And going further, the Wikipedia article references the following paper on patient 0. See if they have a link there. So this was over here. I believe this is the one they referenced in the Wall Street Journal article. So this references an article here. So in hunt for COVID-19 origin, patient 0 points to second Wuhan market. And the man with the first confirmed infection of the new coronavirus told the WHO team that his parents had shopped there. All right. It all sounds like a movie, actually. And continuing further, the World Health Organization's mission to Wuhan said the coronavirus most likely spread naturally to humans through an animal. And this Wall Street Journal's Jeremy Page reports on what scientists learned during this weeks-long investigation. This is Wuhan Institute of Virology, and they're all masked up. And this article is from February 26, 2021. Interesting. So, again, note that his parents, quote, his parents shopped at a Wuhan seafood market, and somehow that makes headline news. Anyways, the quote, virus that is blamed for the above questionable statistics is the severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2 or CoV-2. And here is another Wikipedia link. This is on that quote virus itself. And here's some Electron images of it. Make this a bit smaller. So there is some colorized Electron microscope images of this. So colorized transmission electron micrograph of SARS-CoV-2 or CoV-2 virions, or just virus particles, with visible coronae. So these are these supposed solar corona on the edges there. And yeah, just on these around it like that and so on. Very interesting. Now here is a model of it. So here's model of the external structure of the SARS-CoV-2 virion. So yeah, this is much more detailed cartoon of this colorized. So it's actually black and white, but they just added colors of the supposed different proteins or the genetic material inside. Yeah, it's very interesting. Going further, and now here's transmission of SARS-CoV-1 and 2. 2 is the new one. CoV-1 was in the early 2000s, I believe. So from mammals as biological carriers to humans. So there is bat to raccoon, or bat to human, and there's to human, and then human to others. Yes, this is like Sesame Street. Transmission of SARS-CoV-1 and SARS-CoV-2 from mammals to biological carriers to humans. All right, so there is a bat. So samples taken from Rhinolophus sinicus, a species of horseshoe bats, shown an 80% resemblance to SARS-CoV-2. So supposed samples from it, do some sequencing, and assume there's a virus in this bat, and then somehow they match up with this SARS-CoV-2, or 80% resemblance of it. So yes, poor bat there. Going further, so genomic organization of isolate. So yes, isolate Wuhan. This is Wuhan human 1, the earliest sequenced sample of SARS-CoV-2. And here's some. These are genetic sequences. And this is one string. There's a title for it. And here's another string of genetic sequences, or just base pairs, and so on. And it has about almost 30,000 total. So here's the, this is Genome ID, and this is NCBI, or National Center for Biotechnology Information. Genome ID 86693, and the genome size is 29,903 bases. And year of completion, 2020. Now here is another supposed isolate. So transmission electron micrograph of SARS-CoV-2 virons read, isolated from a patient during the COVID-19 pandemic. So if this is isolated, why is there other stuff? This is colorized, so these little things are supposed to be the virus, and these are other cells. In other words, it's not common sense isolated. It's whatever this is. Going further, and now here is a phylogenetic tree based on whole genome sequences of SARS-CoV-2 and related coronaviruses. So here's SARS-CoV-1, 79% similar to SARS-CoV-2. And there's the SARS-CoV-2 related ones. You have all these other random genetic codes. There's from bats, pangolins, and so on. And you have a lot of these bats. And there's the SARS-CoV-2 one. All right, here's some more digital colorized scanning electron micrographs of SARS-CoV-2 virons, yellow, emerging from human cells, cultured in a laboratory. So you have human cells, and then you put in a petri dish and grow them, add a bunch of other stuff to it, and then you take these electron micrographs images, which are pretty much stationary. There's no actual video, so so they're emerging, but you're not actually seeing anything emerge. You just see these blobs, and then colorize it and say those are the virus or viron. All right, let's continue further, and now we're going to look at ChatGPT, which is a pretty amazing tool, actually. It has some flaws, but it's new, and it's very good at just gathering mainstream information. So ChatGPT on the main paper on COVID virus isolation. So with the rise of artificial intelligence, or AI, and here I put this back in quotations just because, just to be more neutral, because this video is about just analyzing the claim that there is even a virus there. So with the rise of artificial intelligence, AI, it is now quicker to gather and synthesize mainstream information. And I say, quote, mainstream because ChatGPT, like Wikipedia, is biased towards information that is already widely held by the mainstream institutions and public at large. For mainstream information, I mainly cite Wikipedia, but where it comes short, I have found that ChatGPT helps fill in the gaps. For example, in this discussion, ChatGPT gave me the main paper cited on COVID-19 virus isolation. All right, so here if you go to chat.openai.com and basically /chat, then you can sign in with your make an account. I just signed in with my Google account. And here's what the question I asked. So I asked it, this is in March 18th, 2023. So it probably changes up how it answers, but you can get similar stuff like this. Anyways, so what is the question I asked is, what is the main paper on isolation of the virus that causes COVID-19? And ChatGPT states, the main paper on the isolation of the virus that causes COVID-19 is, quote, a novel coronavirus from patients with pneumonia in China 2019 by Zhang et al., published in the New England Journal of Medicine in February 2020. This paper describes the isolation and characterization of the virus from patients with severe respiratory illness in Wuhan, China, and provides key information on the genetic sequence, morphology, and pathogenicity of the virus. The findings of this paper were critical in confirming the causative agent of COVID-19 and have been widely cited in subsequent research and public health efforts related to the pandemic. And then MES asked again, give me a link to the link of that paper. And ChatGPT, certainly, here's a link to that paper, and there's a link. And so ChatGPT is pretty epic for mainstream information. Also, just a note here, they said it published February 2020. It was actually published in January, but in the, I believe, print edition or their routine issues when they post, it was in the February version. But the actual paper was published in January, I believe. Anyways, let's continue further. All right, so here is the paper. So paper, a novel coronavirus from patients with pneumonia in China 2019. And the following is the paper that ChatGPT referenced as the main paper for, quote, COVID virus isolation. Series from the New England Journal of Medicine, nejm.org, and so on. I retrieved this March 18th, 2023. All right, continue further. So here is that article, and here's an editor's note. This article is published on January 24th, 2020 at nejm.org. However, it was part of the February 2020 issue. So it's interesting numbers too. 2020. Lots of twos there. Yeah, so it was published on the February 2020 issue of New England Journal. I believe that's their either print or issue / edition versions. But whereas the actual article was on the website earlier than that, like almost a month earlier. So anyways, this is a novel coronavirus from patients with pneumonia in China 2019. This is from Nazu, and then I'm not going to pronounce these names. Dingyu Zhang and so on, and this is from the China Novel Coronavirus Investigating and Research Team. And note here, it has 17 references, but then it was cited 15,825 times in articles and other papers and so on. It's fascinating stuff. So again, as I just stated it, note that the paper was published on January 24th, 2020, then updated on January 29th. So if you click the PDF version of it, it was actually updated in January 29th, 2020, and it appears in the February 2020 issue of the New England Journal of Medicine, nejm. All right, now let's take a look at the paper abstract or summary. I'm just used to the word abstract. So let's go over the summary or abstract of the paper. And here's the first part. Then I'm going to go over some of the technical terms and then continue back on the paper. So summary. In December 2019, a cluster of patients with pneumonia of unknown cause was linked to a seafood wholesale market in Wuhan, China. A previously unknown beta coronavirus was discovered through the use of unbiased sequencing in samples from patients with pneumonia. All right, so now let's go over some of these terms. So unbiased sequencing refers to genetic sequencing techniques aimed at minimizing any biases or errors in sequencing and data processing. And pneumonia is inflammation of the lungs, primarily the alveoli, and is supposedly caused by, quote, infection with, quote, viruses or bacteria, or less commonly by other microorganisms. And again, just a note here. So this one here, I have links to all these technical terms. This one I use ChatGPT. It's just because the Wikipedia didn't have Yeah, didn't have a complete information on that. And then here's pneumonia. This was from Wikipedia and so on. And I'll just highlight these. Yeah, all of these links are all on there. All right, continuing further. So inflammation is a complex biological response to harmful stimuli and is characterized by heat, pain, redness, swelling, and loss of function. And these are just mainstream explanations of these. And alveoli are millions of tiny air sacs in the lungs where oxygen/CO2 gas exchange occurs, with the outer environment and the blood, and back and forth and so on. And in other words, that's pretty much where the source of breathing comes in, or getting the oxygen from the atmosphere and then exhaling the CO2 waste out of the body. And yeah, so here's this is the trachea or windpipe, and then you have the lungs, and then inside these lungs, you have these. It just branches out over to these bronchi, and then these smaller bronchi, and then or bronchioles, and then they expand outwards to these alveoli. That are these millions of air sacs, and so on, and you connect them to these blood vessels and the outer atmosphere connecting to them. All right, so inflammation of these alveoli inside the lungs is basically considered pneumonia. So they get inflamed, there's a lot of heat, pain, stress, redness, swelling, and so on. And so now let's continue further. So those were the terms there. Continuing further, or actually before I continue further, I just want to add another definition here for beta coronavirus. I'll put that right here. So beta coronaviruses are one of 4 genera or classes of coronaviruses, and includes SARS-CoV-2, the alleged quote virus that quote causes COVID-19. So that's basically a classification of these so-called viruses. Anyways, going further. So continuing further in the paper, in the summary, human airway epithelial cells were used to isolate a novel coronavirus named 2019 nCoV, which formed a clade within the subgenus sarbecovirus, and then this is the inside the orthocoronavirinae subfamily. All right, so different from both MERS-CoV and SARS-CoV. The 2019 nCoV is the 7th member of the family of coronaviruses that infect humans, and enhanced surveillance and further investigation are ongoing. And this paper was funded by the National Key Research and Development Program of China and the National Major Project for Control and Prevention of Infectious Disease in China. Interesting. All right, so now let's continue into more of these technical terms. So epithelial cells right here. So epithelial cells are the cells that line the outer surfaces of organs and blood vessels and the human airways. So this is human airway, nose, mouth, and down to the lungs. These ones have the pseudostratified type of these epithelial cells. And pseudostratified is named such that since they have the appearance of consisting of multiple layers of cells, but are actually just one layer. And here's types of epithelium. So you have the simple squamous, looks like that. Simple cuboidal, looks just like cubes like that. And then simple columnar, so just like columns. And there's a transitional, it's like columns and a cuboidal. There's stratified cuboidal or cuboidal, and then there's stratified squamous. Kind of looks like flat and stratified as well. And then you have the pseudo-stratified columnar or a columnar. So this one's a columns, but then this one looks like it's small. It's stratified with multiple layers, but they're actually just all squished like sideways like that. So they're not actually on top of each other. So yes, that's a pseudo-stratified, and these are the ones that line the nose, mouth, and down to the lungs. And and so here's a graphic of that pseudostratified ciliated columnar epithelium. So there's lots of terms. There's a lining of this throat right there, or the trachea, and then there's the cilia right there. These are fibers, hair-like fibers, and they move up and down the mucus and particles in a coordinated fashion to get them out of the mouth and so on. And there's the nuclei, there's the cells, and there's the cytoplasm, which is all the material inside the cell. And there's a basement membrane, there's a lamina propria, or loose connective tissue, on this side of these organs right there. It's fascinating stuff. And going further, so this forms a clade, or clade, I'll call it clade. And this is just a grouping of organisms that have a common ancestor and contains all of its descendants. For example, the blue and orange groupings are clades, but the green is not a clade. So in other words, this blue contains all of its descendants, so that is a clade. And likewise, the red also contains all of the descendants, that's called a clade. But the green is not, because there is this part right here. So it does not contain this segment here. But then again, you can make the blue and green, that'll be a clade. Or you can make this entire thing, and that'll be a clade as well, because they'll contain all the ancestors. So again, that's just a taxonomical grouping setup. In the subgenus, and this is from this part right here. So these they isolated a new or novel or new coronavirus forms a clade within the subgenus Sarba-coronavirus and this subfamily and so on. So the subgenus, that is so a subgenus is a taxonomical or grouping directly below genus. And for example, the genus Panthera has subgenera, just plural of subgenus, the lion, tiger, jaguar, and leopard. So yes, these are Panthera or Panthera genus, and these are subgenus. Anyways, then the Sarbecovirus, that's short for SARS beta coronavirus, is a subgenus of beta coronavirus. And beta coronavirus is one of 4 genre or plural of genus of coronaviruses. The others are alpha, gamma, delta, and so on. And then orthocoronavirinae is a subfamily in the family of Coronaviridae. And then the term coronavirus refers to any member of the Coronavirinae subfamily. Anyways, that's just a lot of technical taxonomical terms. And just to give you just an overview of how this setup is. So if you have a domain, for example, eukaryotes or multi-celled organisms, and you have a kingdom, for example, animals or Animalia, and then you have this phylum, it's chordata with backbones, class Mammalia, mammals, and then you have order Carnivora or carnivores. You have the family Canidae or canines. You have the genus Vulpes, species, and that is Vulpes. Same thing as red fox. So that's just a taxonomical setup. So now for the coronavirus, the SARS-CoV-2, there's a taxonomical rank for it. So you have unranked is just as virus. Unranked is interesting. And then you have the realm. That's a new one I've heard before. Ribaviria. And kingdom Orthoranviri. Anyways, there's phylum. You have this, and then class, and so in order. Then you have the family is a Coronaviridae. Then you have the genus Beta coronavirus. And then subgenus Sarbecovirus. And then you have the species, which is back to, this would be like the red fox. Then you have the species severe acute respiratory syndrome related viruses. And then the actual virus, severe acute respiratory syndrome coronavirus 2. So a whole bunch of these species there, all related. All right, so that was the abstract. Now let's just summarize it quickly. Again, the main idea of this paper, or this video that I'm reviewing, is going to be on the virus isolation, but I wanted to go through the entire paper in detail first. So summary of the abstract. Let's go over a quick summary of the abstract. # summary of the summary. So patients with pneumonia or inflammation of the lungs of unknown cause was, quote, linked to a seafood market in Wuhan, China in December 2019. And then genetic sequencing of samples from pneumonia patients was used to, quote, discover an unknown coronavirus. And then human airway epithelial cells, or cells that line the nose, mouth, and down to the lungs, were used to, quote, isolate a novel or new coronavirus named 2019 nCoV. All right, so to break it down even further, so basically they found some patients with inflammation of the lungs. Then they did genetic sequencing to discover a new virus, and then they somehow isolated it using the cells that line the nose, mouth, and lungs, and so on. So yes, very interesting. So let's we're going to look through the methods and so on to see if this all makes sense. All right, so now that we've covered the abstract or summary, let's take a look at the next section of the paper, and that's the paper introduction. And a lot of this is going to be on just the mainstream narratives of virology in general. So let's go over the paper's introduction. So emerging and re-emerging pathogens are global challenges for public health. And coronaviruses are enveloped RNA viruses that are distributed broadly among humans, other mammals, and birds that cause respiratory, enteric, hepatic, and neurological diseases. Yes, that's a big claim there. But again, these are all just assumed in this paper. So a pathogen is a microorganism or agent that can supposedly produce disease. And a viral envelope, for this coronavirus has an envelope, is the outermost layer of many types of viruses and helps to protect the genetic material when traveling between host cells. And respiratory diseases affect the breathing organs, and enteric diseases affect the intestines or gut. And hepatic diseases affect the liver, which is responsible for detoxification, metabolism, and storage of nutrients. And neurological diseases affect the brain, spinal cord, and peripheral nerves, which extend from the brain and spinal cord to the rest of the body. And detoxification here, I bolded these because I'm going to expand on these. Detoxification is the removal of toxic substances from the body, and metabolism is all the chemical processes that occur to maintain life, such as those involved in converting food into energy, building and repairing cells, and elimination of metabolic waste. And a nerve is a cable-like bundle of fibers, or fiber that transmit electrical impulses. And pretty much, controls the body and so on, or sends signals to cause it to move. All right, let's continue further in the paper. So, 6 coronavirus species are known to cause human disease, and 4 of these viruses, 229E, OC43, NL63, and HKU1, are prevalent and typically cause common cold symptoms in immunocompetent individuals. In other words, these are just healthy individuals. You get these common cold symptoms. And let's just go a background on this common cold and so on. So first off, immunocompetence is the ability to have a normal, quote, immune response following exposure to an antigen. And antigens are, or I'll just fix the grammar. An antigen is any molecule or foreign matter that can bind to a specific antibody and can trigger an, quote, immune response. And antibodies are proteins that identify and neutralize, or, quote, identify and neutralize foreign objects such as pathogenic bacteria and, quote, viruses. And an immune response is the body's complex and coordinated response to, quote, invading pathogens or foreign substances that may harm the body. And this common cold, so the common cold is a, quote, viral infectious disease of the upper respiratory tract that mainly affects the respiratory mucosa, basically another word for respiratory epithelium, of the nose, throat, and sinuses and larynx. And symptoms of the common cold include coughing, sore throat, runny nose, sneezing, headache, and fever. And fever is defined as having a body temperature above normal range, and it's typically between 37.2 and 38.3 degrees Celsius, or 99 and 100.9 degrees Fahrenheit in humans. And the upper respiratory tract, this part right here. Yeah, this upper respiratory tract, this part right here. So common cold is viral infectious disease of this upper respiratory tract. And the upper respiratory tract is a region involved in the initial pathway for air to enter the body. And the larynx or voice box is in the top of the neck, involved in breathing, producing sound, and protecting the trachea from food getting inside. And the sinuses are air-filled cavities in the skull connected to the nasal cavity with possible functions in moistening / filtering air and providing resonance in speech. And here's this image of this upper respiratory tract. There's a nasal cavity there. There's a pharynx right there. Then there's the larynx right there, the voice box. And the pharynx is over here. And yeah, this is just behind the mouth over there, and there's the larynx, the voice box, and so on. And there's the lower respiratory tract. There's a trachea, it goes all the way down here, or the windpipe. And there is the the lungs and so on. There's the bronchi that branches out. All right, now let's continue further in the paper. So the two other strains. So remember, here the paper states that there were 6 coronavirus species known to cause human disease, 4 just for the common cold. And now there's 2 other strains, severe acute respiratory syndrome coronavirus SARS-CoV and Middle East respiratory syndrome coronavirus MERS-CoV are zoonotic in origin and have been linked to sometimes, so 2 disqualifiers are linked to sometimes fatal illness. SARS-CoV, now SARS-CoV-1, because the new one is the COVID one, is a, quote, strain or variant of a coronavirus that causes, quote, severe acute respiratory syndrome or SARS, and the word acute in here refers to the sudden onset and rapid progression of symptoms, and syndrome refers to a collection of symptoms associated with a disease or disorder. And a zoonotic disease is an quote infectious disease caused by a pathogen that, quote, jumps from a non-human to a human and vice versa, or non-human animal. I think that's probably more applicable, and vice versa. Yeah, so interesting there. So this one here, these ones supposedly jumped from an animal to humans and so on. So continuing further in the paper, SARS-CoV was the causal agent, or the SARS-CoV-1 was a causal agent of the severe acute respiratory syndrome outbreaks in 2002 and 2003 in Guangdong Province, China. And MERS-CoV was the pathogen responsible for severe respiratory disease outbreaks in 2012 in the Middle East. And given the high prevalence and wide distribution of coronaviruses, the large genetic diversity and frequent recombination of their genomes, and increasing human-to-animal interface activities, or human-animal interface activities, novel or new coronaviruses are likely to emerge periodically in humans owing to frequent cross-species infections and occasional spillover events. So yes, that's the introduction, part of the introduction of the paper. And again, just a lot of mainstream assumptions that this paper just assumes. All right, so genetic recombination is the exchange of genetic material between different organisms, resulting in the production of offspring with combinations of traits different from those found in either parent. And one example is just sexual reproduction in humans, for example. Where the offspring may get traits that are different from both parents. For example, if both are short, and then one could be tall, and that could be, just due to genetic mix-up and so on, just shuffling and exchange. All right, so continue further. So, quote, viruses recombine genomes whenever 2+ quote viral genomes are present in the same host cell. And I haven't actually gone over the mainstream definition of viruses, but I'll go over it right here. So a quote virus is an infectious or quote infectious particle too small to be seen by an optical light microscope, and can only replicate inside the living cells of an organism. And yes, some of them are said to be cause disease or health issues or death in humans and so on. So going further, so spillover events right here. So occasional spillover. Spillover events occurs when a population with a high pathogen prevalence, termed a reservoir population, or occurrence. Actually, I'll delete this occurrence. Basically saying the word occurrence. So with a high pathogen prevalence or occurrence, and this is termed a reservoir population, comes into contact with a new host population, and the pathogen may or may not be transmitted within the new host. And going further, so climate change is said to increase the risk of viral spillover. Yes, if you go in the Wikipedia article on this, saying climate change is going to increase this this basically pathogen swapping into new host and so on. And they may or may not be transmitted within it, but assuming there's going to be another pandemic declared, saying that climate change caused another quote pandemic. Anyways, Continuing further in the paper, in late December 2019, several local health facilities reported clusters of patients with pneumonia of unknown cause that were epidemiologically. All right, I'll try that again. So that were epidemiologically linked to a seafood and wet animal wholesale market in Wuhan, Hubei province in China. And on December 31st, 2019, the Chinese Center for Disease Control and Prevention, or China CDC, dispatched a rapid response team to accompany Hubei province and Wuhan city health authorities and to conduct an epidemiologic and etiologic, basically looking for the cause of disease, investigation. And this paper right here is the report of those findings. So we report the results of this investigation, identifying the source of the pneumonia clusters, and describe a novel coronavirus detected in patients with pneumonia whose specimens were tested by the China CDC at an early stage of the outbreak. And we also describe clinical features of the pneumonia in 2 of these patients. All right. Epidemiology is the study of the distribution patterns, risk factors, and disease conditions in a population. And a wet animal wholesale market is a market that sells live animals or animals butchered when sold. And wholesale refers to selling in bulk and typically to other businesses. And clinical features right here is just observable or measurable. Clinical features are observable or measurable characteristics associated with a disease or patient. Or patients such as symptoms and laboratory findings. All right, that's very interesting. So now let's take a look at the summary of the introduction. Yeah, so let's go over a summary of the paper's introduction. So supposedly there are, quote, global challenges of, quote, disease-causing microbes or microorganisms. And coronaviruses have an outer layer protecting RNA inside. And coronaviruses supposedly cause diseases relating to breathing, the gut, liver, brain, and nervous system. And 6 known coronaviruses are quote, or is it double known? So 6 coronaviruses are, quote, known to cause human disease. 4, quote, viruses cause the common cold. And 2 other, quote, strains have been linked to fatal illness. And this is the SARS-CoV or CoV1 caused outbreaks in 2002 and 2003 in China. And the MERS-CoV, quote, caused outbreaks in 2012 in the Middle East. And new or novel coronaviruses are, quote, likely to emerge in humans because coronaviruses are widespread, genetically diverse, and interactions between humans and animals are increasing. And on December 29, 2019, clusters of patients with inflammation of the lungs, aka pneumonia of, quote, unknown cause, were, quote, linked. Again, a lot of these words here, quotations that I put just that were questionable, like what do you mean by linked and so on. Linked to an animal market in Wuhan, China. On December 31st, 2019, the China CDC sent a rapid response team to Wuhan to investigate. And this paper reports the results of the investigation, so identifies the source of the pneumonia clusters, claims to detect a, quote, novel coronavirus in the pneumonia patients that were tested at an early stage of the outbreak. Again, it's all quotations, early stage of the outbreak. Clinical features, aka symptoms and laboratory results, are presented for 2 of the pneumonia patients. Alright, so now the next part of this video, before I get to the paper, I'm going to go over a quick overview of nucleic acids and genomes, because this will serve as good background for the rest of the paper. So before I go further into the paper, let's first go over an overview of the mainstream explanations of nucleic acids, genomes, and cell theory in general. So nucleic acids are chemical compounds essential to all known forms of life. They're composed of nucleotides, which are made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base, also called nucleobase. The sugar plus phosphate make up the helical backbone of this chemical compound. The specific sequence of nucleobases, CGAU in RNA and CGAT in DNA, make up the, quote, genetic codes, basically sequences of these nucleobases, which is these nitrogenous bases. And base pairs, or BP, are two nucleobases bonded together and form the building blocks of the DNA double helix. And deoxyribonucleic acid, or DNA, is made up of two polynucleotides, or just multiple nucleotides combined together, that coil around each other to form a double helix. And DNA contains genetic instructions for the development, functioning, growth, and reproduction, put right here, of all known organisms and many, quote, viruses. And ribonucleic acid, or RNA, is typically a single-stranded polynucleotide and typically folded onto itself in nature. And then RNA plays roles in coding, decoding, regulation, and expression of, quote, genes. And many viruses, many, quote, viruses encode their genetic information using an RNA genome. And a genome is all the genetic information of an organism and consists of the nucleotide sequences. It consists of all the nucleotide sequences. These are still typically just labeled by the base pairs and so on, or just the bases right there. Going further, the human genome consists of 23 pairs of chromosomes, making 46 total. So there's pairs 23 times 2, 46, including 2 sex chromosomes, which determine the sex of the individual. So the X and Y chromosomes. So chromosomes are long DNA molecules that contain part or all of the genetic material in an organism, and cells split and duplicate the chromosomes during cell division, and so on. So here are the RNA, ribonucleic acid. Looks like this. There's a helix of the sugar phosphate backbone. And then the DNA is deoxyribonucleic acid. So again, very similar. This one, I believe deoxy is when oxygen is removed. It's deoxy. I believe that's what it is. Anyway, so then these are the nucleobases right here, and these are in the different colors right here. So this is adenine, is this right here, it's yellow. These yellows connect here with this red, thymine, and so on. And if you take a look at the RNA and DNA, so RNA has the cytosine, the same thing. These line up, so there's the G, so the guanine, adenine, and then the only difference is T-thymine for DNA, and then uracil, or U, for RNA. So instead of that, they'll have this one right here is this U is an orange, whereas this one has the red. Yeah, it's very, very fascinating. And then here's just a quick overview schematic. This is a source typical human cell showing the nuclear genome inside a cell's nucleus, as well as the mitochondrial genome. This is inside the cell's mitochondria, which is the source of the cell's energy production. Yeah, so most of the genetic code is inside this, the nuclei. So here's a cell right here. As for example, here is 2 chromosomes, and they just they duplicate and split up, and then you get 2 cells like that in cell division. And that is one of the chromosome pairs. And these are a whole bunch of these. There's 23 in humans, and then there's the Y is inside. It's a YX. When there's a Y and an X, that's a man. When there's an XX, that is a woman. And then here is the mitochondria. So there is that's a cell. So inside the nucleus, all of these 23 chromosomes are inside this nucleus. Inside the nuclei right there, or nucleus. And then inside this mitochondria is these extra genetic codes in there. But again, most of it is all inside here. All these inside, and they all just divide. And going further, here is a part of the DNA sequence of a, quote, virus genome. And again, it's just all these different nucleobase pairs, right, or bases. Yeah, and this is all labeling it. So this is this would be if you look at the RNA or DNA, this is T, this is U. So this one's a T. So this is I would assume a DNA, yes, a DNA sequence of a DNA virus. So this one is a quote virus that has DNA as genetic code, I assume, and then as opposed to the RNA one, such as the COVID virus. So the human genome has 3. This is 3 billion base pairs. So this is already base. 3,117,275,501 base pairs. This includes 16,569 base pairs for the human mitochondrial genome, And note that the quote, the COVID quote virus genome has 29,903 base pairs. So compare to that 29,000, then you got 3 billion for humans. And going further, so cell theory has 3 tenets or beliefs. All cells, all living organisms are composed of 1 or more cells. The cell is the basic unit of structure and organization in organisms, and cells arise from pre-existing cells. So cell theory was universally accepted, but now as some biologists consider non-cellular entities such as quote viruses as living organisms, which means one tenet is disputed. So this right here, all living organisms are composed of 1 plus cell. So again, this is questioned because the theory of viruses is that they exist even without this cellular setup, because they're essentially just a little particle with genetic code contained within. And so a lot of people aren't sure what viruses are living or not living, and so on. Again, based on how the cell theory is presented. Going further, the central dogma of molecular biology states that genetic information flows between DNA and RNA to protein, but not from protein to RNA. So if you have DNA, right here, so DNA you could replicate to get DNA again. So you can go DNA to DNA. You can also go DNA to RNA, and then you can go from RNA to go back. This would use a reverse transcription enzyme and so on. And then you can go RNA to RNA, and these in blue is just a general. So this is generally what happens. And then the special type, you can go backwards in special forms, or RNA to RNA. And then this will just go generally just goes to a protein and build the entire cell, just the organism. And here's another more complete diagram right here. So you have this DNA. It can replicate using this DNA polymerase. And a polymerase is an enzyme that synthesizes long chains of polymers or nucleic acids. And enzymes are proteins that act as biological catalysts to accelerate chemical reactions. And these are polymers. So polymers are very large molecules composed of many repeating subunits, and proteins are a very important type of polymer. All right, so so going back right here. So if you go from RNA to DNA, you're going to need the reverse transcriptase. But if you go from DNA to RNA, you're going to need the RNA polymerase. All these are just different enzymes. And then you can also get RNA to RNA using this RNA-dependent RNA polymerase. You can go backwards right here for the different sense. We won't go over the sense, but basically a different form of that sequence. Or which one's used as template and so on. And here I actually just added just a definition for sense RNA. So our sense RNA refers to complementary RNA sequences, and positive sense RNA can be directly translated to protein, as this image shows. If it's plus sense, can go right here. But then if you replicate this, is typically you would have another nucleotide sequence, and it'll match up on this. But then it's going to be everything's going to be on the opposite side, and then you replicate it over here by matching up these sequences on top, these base pairs. And then you're going to have one like this, but this can't be translated directly to protein, so you're going to need to flip it over using this RNA-dependent RNA polymerase to get the other section over here, and then you could go to the protein. And again, it's just simplified overview of it. And then there's translation. This is RNA to protein via the ribosomes inside the cell. That is the place that supposedly produces the protein. All right, and now that we have gone over this overview, this quick one, now let's get back to the paper and now look at the paper's methods. And the first part is on detecting, quote, viruses. So now let's get back to the paper and take a look at the paper's methods. The first of which involves trying to, quote, detect known viruses. So methods, viral diagnostic methods. So 4 lower respiratory tract samples, including bronchoalveolar, or let's try that again. So including bronchoalveolar lavage fluid, were collected from patients with pneumonia of unknown cause who were identified in Wuhan on December 21st, 2019, or later, and had been present at the Wuhan Wuhan seafood market close to the time of their clinical presentation. And 7 bronchoalveolar lavage fluid specimens were collected from patients in Beijing hospitals with pneumonia of known cause to serve as control samples. Again, this paper doesn't actually go through much more information on these supposedly controlled samples. So the lower respiratory tract is the lower part of the groups of organs involved in moving oxygen in and out of the body. And then bronchoalveolar lavage is a method in which a tube is inserted into the lower respiratory system and fluid introduced, then collected for examination. So if you have this right here, so there's a lower respiratory tract. So you put a tube inside, and then you pump in some water inside there, all the way inside, and then you would extract this fluid out. And that's the bronchoalveolar lavage fluid there. So you pump it all the way inside here, and then you get the fluid out. Just a pretty invasive technique there. Continuing further. So let's continue further in the paper. So extraction of nucleic acids from clinical samples, including uninfected cultures that served as negative controls. Again, there's very little info on these supposed negative controls in this paper. So was performed with a high pure viral nucleic acid kit as described by the manufacturer Roche. And I'm not going to go over though, but just the results or supposed results of. So this were cultures right here. So including unaffected cultures. So they would extract these from these supposed clinical samples. So a cell culture is a process of growing cells under controlled conditions. And a negative control is a condition expected to produce a negative result, such as giving samples without the active ingredient being tested. And here's a cell culture plate or quote petri dish, like that, just a plastic or glass case like that, dish. I mean, and now here's epithelial cells in culture stained for keratin, which is a structural protein in red, and DNA in green. So these epithelial cells. Like that again. Hard to get too much detail on this, but it's interesting nonetheless. So continuing further, so extracted nucleic acid samples were tested for viruses and bacteria by polymerase chain reaction or PCR using the RespiFinder Smart 22 kit or Pathfinder BV and the LightCycler 480 real-time PCR system in accordance with manufacturer instructions. And samples were analyzed for 22 pathogens, 18 viruses, and 4 bacteria, as detailed in the supplementary appendix. And this appendix just gives you the sequences or the list of these supposed pathogens and viruses that they were looking for, and bacteria. So the polymerase chain reaction, or PCR, is a method of rapidly making millions to billions of complete or partial copies of a specific sample of DNA. And PCR cycles refer to the repeating number of steps during DNA replication. So if you have a DNA template with sequence of interest, so you have this like this, a sequence, and then what you also have is a whole bunch of these supposed primers, or these starting sequences, or these target ones. Then this this DNDP or deoxyribose nucleoside triphosphate, or DNDPs, are the building blocks of DNA that are used by DNA polymerase enzymes to synthesize a new DNA strand. So these are the building blocks of the DNA, a whole bunch of these compounds, and you have the polymerase, which is the enzyme that builds those up or makes the DNA. You have these primers, or these starting sequences, and so on. So denaturation. So if you have this target DNA template, denature it, you'll just separate these. So it usually heats them up, separates it, and then this is annealing. So the next step is that this target primer connects to them. This connects here, and then these are the building blocks of it. With this polymerase starts adding these, and then you can form a template like this, and I believe that they'll be matching up. That would match up like this, following this primer set. This should be a target of that. Yes, I'm not going to go into too much detail of that. And then we'll do it on this in this case, because you separate each strand. Now this builds it up using these building blocks, and that primer initial setup, and then you do the same thing again. Then you would second cycle, you're going to denature, and then anneal, and so on. So each time you get double. Then the third cycle you double, and then the nth, and then it goes to copies are 2 to the power of n. You get a PCR product, and so on. So you basically amplify this to millions and billions of copies, and so on. So deoxyribose, actually we went over those. So now let's continue further. And here, actually, before we go further, I just want to add this primer definition. So a primer is a short, single-stranded nucleic acid used by all living organisms in the initiation of DNA synthesis. Interesting. So that's the first one. And then it's going to synthesize the remainder. So that's the start, and then it gets all these and matches up according to this sequence, or the likely sequence that it wants to get to. It's very interesting. So now let's continue further. So in addition, unbiased, high-throughput sequencing described previously, it was used to discover microbial sequences not identified by the means described above, and a real-time reverse transcription PCR or RT-PCR assay was used to detect viral RNA by targeting a consensus RD-RP region of pan, this beta COV, as described in the supplementary appendix. And let's go over some more technical terms. So high throughput sequencing, or second generation sequencing, or next generation sequencing, NGS, refers to advancements in sequencing technology that allows for entire genomes to be sequenced at once. And the term unbiased refers to the method where specific sequences are not preferentially amplified or sequenced over others. And here's just the history of sequencing technology. There's Sanger method, human mitochondrial genome sequence, 1981. Human Genome Project, 1990. Then 1985, complete cell genome. Then 1986, complete eukaryotic genome. I think that's multicellular genomes. And then 2001, complete the Human Genome Project. And then second generation sequencer right here. 2005, then second generation sequencer, genetic analyzer 2. So these ones do basically in bulk. And there's a research human microbiome project. There's all the microorganisms in the body. And then there's a third generation sequencer, PacBio-RS. There's a nanospace sequencing. And there's a third stage human microbiome project. Again, this is getting pretty advanced stuff. So real-time polymerase chain reaction, or real-time PCR or qPCR monitors the replication of a targeted DNA molecule during the PCR in real time and not at its end as in conventional PCR. And reverse transcription PCR or RT-PCR combines the reverse transcription of RNA into DNA, and then it replicates the DNA targets using PCR, which is, again, split the strands, and then you would then replicate and split and replicate. And reverse transcription is the process in which the enzyme reverse transcriptase is used to generate DNA from an RNA template. In an assay over here, because it's using this RT-PCR assay. An assay is an investigative procedure to detect or measure a specific biological entity or function. Basically, just a procedure list. And then the last one right here, so they use this RT-PCR to detect viral RNA by targeting a consensus RD-RP region of this pan-B-CoV. Let's see what those all mean. So pan-beta-CoV refers to a group or test designed to detect a broad range of beta coronaviruses. And RNA right here, this RD-RP, R is just stands for RNA-dependent RNA polymerase. RD-RP, or RNA replicase, is an enzyme that catalyzes the replication of RNA from an RNA template. So this replication is RNA-dependent RNA polymerase. I believe we saw that in the in over here. So this is the central dogma of molecular biology, and this is the RNA-dependent. So RD RNA polymerase, RD-RP. So you need that enzyme to go in replication, either go from here or going back, and so on. Different sense. All right, that's very fascinating stuff. All right, so that is the so RDRP is the replicase is the enzyme that catalyzes the replication of RNA from RNA template. And the RDRP region refers to a specific region of the genome of RNA code viruses that encodes for this enzyme, this RDRP enzyme. And the consensus right here, so it's targeting a consensus RDRP region of these coronaviruses. And then so the consensus RDRP region refers to a segment of the RDRP gene that is highly conserved among different quote strains or isolates of a quote virus species. All right, it's very, very fascinating stuff. So now let's take a look at a summary of detecting the quote virus. So let's quickly summarize the quote virus detection methodology in this paper. So 4 samples were taken from patients with pneumonia of unknown cause in Wuhan on December 21, 2019 or later. And 7 samples were taken from patients with pneumonia of known cause in Beijing to serve as control samples. And nucleic acids were extracted from the clinical samples and, quote, uninfected cultures. PCR or DNA replication tests were done on the extracted nucleic acids to test for 22 quote viruses and 4 bacteria. And here's one to add the samples right here. So this is a lower respiratory samples. Yes, so the 4 respiratory samples. Same with this one here. 4 lower respiratory samples. And I believe this is a fluid. We're going back here. So 4 viral diagnostic methods. So 4 lower respiratory tract samples, including bronchoalveolar lavage fluid, and so that's including that. I don't know what it means. Is it all of them that or different? Then while this one right here, 7 this fluid samples right there for the known cause of pneumonia. So I'm just going to say. I'm going to assume 4 low respiratory fluid samples. And then the same one right here. Fluid samples were taken from patients with pneumonia and so on. So anyways, and then they were tested for these quote viruses. And I would assume that the known cause ones didn't, they would test for these, or at least some of these that they're being tested for. But then the unknown one wouldn't be testing for these known viruses and bacteria. So high throughput sequencing were used to discover microbial sequences, looking for these molecules and not the actual quote virus, not identifiable by the above means, which we're testing for. Testing for the known sequences, and also using PCR and so on. And going further, so RT-PCR, so again, this is, the PCR is DNA replication. Then RT-PCR is RNA conversion to DNA, then DNA replication. Assay was used to detect, quote, viral RNA by targeting a consensus RDRP region of a group of beta coronaviruses. And this is the genera of coronaviruses that includes the alleged COVID-19, quote, virus titled SARS-CoV-2. Alright, so now let's take a look at the next section, and this is on the paper methods, and it's going to be on, the first part of it is going to be on the isolating the, quote, virus. So now let's take a look at the next section in the above paper, which is on allegedly isolating the, quote, virus, and this paper starts off over here. So, isolation of virus, bronchoalveolar lavage fluid samples were collected in sterile cups to which virus transport medium was added. All right, and remember that this bronchoalveolar lavage fluid is they just put a tube down the throat and put a fluid inside, and then extract that fluid and all the material inside in the lower respiratory tract. All right, so they put this in sterile cups to which virus transport medium was added. And so virus or viral transport medium, VTM, is a chemical preservative used to preserve, quote, virus specimens during transport instead of using cumbersome supercooling equipment. And typical VDM components may include saline solution, just a salt solution, phosphate buffered saline PBS, or fetal bovine serum FBS. Yes, that's interesting. A buffer solution is an acid or base aqueous solution used to maintain a constant pH when a small amount of strong acid or base is added to it. And acids and bases are defined as in the three theories shown in the image below. So here's classification of acids. There's three different theories of them, and which some of the theories overlap with the other ones here. So classification of acids and bases. So acids, you'll have a Lewis acid accepts electron pair. Then Bronsted-Lowry acid donates proton or H+. And then Arrhenius acid dissociates into H+ when dissolved in water. And then classification of bases, you have the opposite. Lewis base donates electron instead of accept. This one's accept, this one's donate. Then Bronsted-Lowry base accepts this proton. So this one is, accepts it, whereas this one donates this electron pair over to this. And then the Arrhenius base dissociates into OH- when dissolved in water. So this goes to OH. The other one goes into plus. So acids go into plus in water. This one OH- dissociates into that. All right. Let's go back up here. All right, and now the pH or potential or power of hydrogen indicates the acidity or basicity of an aqueous solution. The higher the value, the more basic, and the lower, the more acidic. And an aqueous solution is a solution in which a solvent is water. And a solvent is a substance that dissolves another substance called a solute or solute, and resulting in a solution. And then this, lastly, this fetal bovine serum, FBS, is extracted from blood. Is extracted from blood drawn from a bovine or cow, bull, or oxen fetus after it is killed at a slaughterhouse. That's pretty wild. And then serum is the part of the blood not involved in blood clotting. So they would extract that serum from this fetal bovine, or this fetus from this cow, bull, or oxen that's killed at a slaughterhouse, and then mixed in with this, and may oftentimes be used as this viral transport medium. So then they would put these samples inside this serum from this slaughtered fetus. All right, so that's quite baffling. Continuing further in the paper, samples were then centrifuged to remove cellular debris, and the supernatant was inoculated on human airway epithelial cells, which had been obtained from airway specimens resected from patients undergoing surgery for lung cancer, and were confirmed to be special pathogen-free by NGS. All right, a lot of steps in this quote virus isolation. So centrifugation involves rapid spinning to separate objects by density, and the supernatant or supernate is the remaining liquid after centrifugation or settling. Inoculation is the introducing of microbes into the sample. In other words, you're, quote, infecting this sample. Resection is the removal of all or a key part of an internal organ or body part. Again, this is just terms for surgery in general. Cancer refers to abnormal cell growth with the potential to, quote, invade or, quote, spread in the body. And specific pathogen-free, SPF, refers to being free of specific pathogens. And lastly, this NGS, that actually is, let's put this in bracket right here, next generation sequencing. Yeah, so in other words, they just did a genetic sequencing, and then they said, oh, all right, there's no special pathogens, or these specific ones that they're looking for, that may interfere with this test and so on. So going back to that supernate or supernatant, so if you have a solution right here, has mixed in with solvent, and there's a solute, these particles inside. And now, this is a solution. Yeah, the solution is all blended in perfectly inside, but then here is a suspension, is when you can see these visible. And then when you have the supernatant is once you, for example, you centrifuge it or you let it settle, then you're going to have the fluid on top. That's a supernate or supernatant. Yes, and the centrifuge is basically a device for separating particles from a solution according to their size and shape and density, viscosity of the medium, and rotor speed. It just spins it, and then the heavy stuff goes thrown down at the bottom. So you spin this, the heavy stuff goes further out, the more momentum, and the lighter stuff gets closer in. That's spun like that, and then this one is centrifuge, where this would be spinning around. Whoops. Yeah, whoops. This would be spinning around like that, and then the heavy stuff goes down at the bottom. So large size or heavy stuff, and then medium size, small. Then there's a solvent. It gets separated, and you'll have a supernatant of small particles and the solvent. All right, that's schematic of centrifugation. And now continuing further in the paper, so human airway epithelial cells were expanded on plastic substrate to generate passage 1 cells and were subsequently plated at a density of 2.5 times 10 to the power of 5 cells per well on permeable transwell COL or 12 millimeter diameter supports. All right, so let's make sense of all this. So plastic substrate right here refers to a plastic surface on which cells are grown. And expanding cells means growing cells outside of their natural environment. And passage 1 cells refer to cells grown and expanded in culture for the first time after obtaining from a primary cell source, such as a tissue organ. And plating refers to growing cells or cell dishes. Growing cells on dishes or multi-weld plates, which are used. Let's write multi-weld plates, which are used to produce more consistent and reproducible population of cells. So multi-welds just have holes in them or tubes, very, very tiny. Yeah. So here is a transwell coal support. Looks like this, and so you could have this would be to hold it and so on, but then there'll be these super tiny ones there. And they look like this. Continuing further, so here's some of these plastic substrates. And now here's a schematic of a transwell insert. It's gonna look something like this. You have the upper compartment. You have the transwell insert. I believe that's these handles there. Or this whole thing's insert. So yeah, this whole thing's insert. All right. So now you have this upper one. Now you have the microporous membrane. So those are the wells. So you have wells inside there. And there's the lower compartment and so on. So, and this would be, so it looks like that. So you have a container there that can have the solvent or some liquid material or medium. And and then the inserts go inside. So yes, and you can grow whatever you're trying to grow. And these are all cells. And you can grow them more consistently. It's very, very fascinating. And again, the number of cells right here, 2.5 times 10 to the power of 5. That's about, that'll be 3 would be 100,000. This would be, 3 would be 1,000. Then add another 210. So this is about 250,000. Yeah, so absolutely dense right there. 250,000 cells. Very, very interesting. And now continuing further. Yeah, further in the paper. So human airway epithelial cell cultures were generated in an air-liquid interface for 4 to 6 weeks to form well-differentiated, polarized cultures resembling in vivo pseudostratified muscular. All right, let's try that again. Mucociliary epithelium. All right, so let's make sense of all this. So air-liquid interface cell culture, or ALI, involves growing cells in which the bottom portion are in contact with a liquid medium and the top exposed to air. And this is to better mimic in vivo conditions. And in vivo is Latin, or derived from the word that means, or the words that mean within the living, and refers to tests done on the natural living organisms. In contrast, in vitro, Latin for within the glass, refers to tests done in a laboratory setting outside of normal biological conditions. And cellular differentiation is the process. Yeah, and this is dealing with this right here, well differentiated right here. I'll just bold this. Is the process of cellular differentiation is the process in which one cell changes from one type to another, and stem cells can differentiate into various cell types. And the further that cells differentiate, the more specific type of cell they become. So you have stem cells becoming different cells, and then those ones become more specific types of cells. And cell polarity refers to spatial differences in shape, structure, and function within a cell. So you'll have a difference on one side than the other, and so on. And mucociliary refers to the combination of mucus and cilia in the respiratory system. And mucus is a sticky substance. Is a sticky substance that helps trap foreign particles, and cilia are hair-like structures on the surface of epithelial cells and beat in coordinated manner to move the mucus and trap particles up and down, up and out of the respiratory tract, as I showed earlier. And here, just teleported up above here. So there's a cilia right here. It moves in coordinated up and down, and it moves the it moves particles out there. And then usually the mucus traps particles, and then this moves it, and so on, out of the respiratory tract. All right, continuing further. So now here's a schematic of submerged A. This one is completely all liquid, and air-liquid interface B. This is B. Culture of primary human bronchial epithelial cells, HBEC, grown using porous culture inserts. So you have those inserts that I showed earlier. So here is primary HBECs. These are just the human bronchial, again, inside the lungs. Epithelial cells, you can grow them like this in a liquid medium, or you can have this pseudostratified epithelium grown with this air interface set up like that. There's that pseudostratified type. Yeah, so some of it, this is the liquid, and then the top is exposed to air. And there's the porous inserts to get those nutrients and so on to help grow. All right, very fascinating stuff. And here is a schematic of stem cell differentiation into various tissue types. You have a stem cell, it's undifferentiated, and then becomes, for example, a muscle cell. And this one can't become a fat cell, can't become a sex cell, and so on, because it's become differentiated. But only this cell can. So yes, if stem cell goes to here, and then or become fat cell, bone cell, blood cell, nervous cell, and so on. Immune cell, epithelial cell, and then the sex cells over there. And going further here's another schematic of that, of the respiratory epithelium. So that's lining around this lower respiratory tract and the upper as well. So movement of mucus to the pharynx to get to this mouth, and you can spit it out. And it goes up and down with the cilia, and there's a particulate gets trapped in it, and this sticky substance is the mucus around it. All right, very interesting. As a mucus layer, cilia, and then a ciliated columnar epithelial cell, and then you'll have the lamina propria, or loose connective tissue, and basement membrane underneath it. This over here, underneath the cells. All right, so illustration depicting the respiratory epithelium. Very interesting. All right, so now let's continue further in the paper. So prior to infection, apical surfaces, or the top parts of them, of the surfaces of the human airway epithelial cells were washed 3 times with phosphate buffered saline, and then 150 microliters of supernatant from bronchoalveolar lavage fluid samples was inoculated onto the apical surface of the cell culture. So you have the cell cultures, they get washed 3 times with this phosphate buffered saline solutions, and then you have this 150 microliters of the supernatant from those airway fluid samples, and then you would quote inoculate or quote infect those cell cultures. All right, so again, everything's just assumed it's an infection. Apical refers to the top part of the cells which extend into the open airway. And over here, a microliter, or UL, this is a microliter symbol right there, is just 10 to the negative 6 liters, which is equivalent to a cubic millimeter or millimeter cubed. And you could draw it. If a millimeter is about this let's see, this is like more than a millimeter. See how big a millimeter would be. So this is several millimeters right here. Is a microliter, like that. And here we have 150 of those. Yeah, so if you do that, it would probably be like, let's say, something like this, I would assume. This amount of fluid or less. Yeah. Something like that. And 150 microliters is equal to 0.15 milliliters. Yeah, so even smaller than that, maybe. It's just a little tiny puddle. like that maybe is more likely. All right, so MES note, how does the paper know there is a quote virus in which they are quote infecting the cell cultures with? So they just take from these fluid samples, they take the supernatant, and then they just place it onto these cultures. Yeah, so then the apical surface of these cultures. So they make the cultures, and then they would take the supernatant and then inoculate onto the top of the cell cultures. How do they know? That's a good question. All right, so now continuing further on the paper. So after a 2-hour incubation at 37 degrees Celsius, unbound virus was removed by washing with 500 microliters. And note here the paper uses lowercase l, but I'm used to the uppercase L. And here I actually changed it for them here. I'll put it back here. So they use UL like that. I'm just used to writing microliter and capital L always. So yes, that's just a way I've been working with. But yeah, so they mean the same thing. So yeah, so after a 2-hour incubation at 37 degrees, unbound virus was removed by washing with 500 microliters of phosphate buffer saline for 10 minutes. So you're washing it for 10 minutes after you grow this for 2 hours. And then human airway epithelial cells were maintained in an air-liquid interface incubated at 37 degrees with 5% carbon dioxide. And then every 48 hours, 150 microliters of phosphate buffered saline was applied to the apical surfaces of the human airway epithelial cells. And after 10 minutes of incubation at 37 degrees Celsius, the samples were harvested. And then pseudostratified mucociliary epithelium cells. Yeah, so pseudostratified mucociliary epithelium cells were maintained in this environment, and apical samples were passaged to a 1:3 diluted or 1:3 diluted vial stock to new cells. All right, let's make sense of all this now. An incubator maintains optimal temperature, humidity, and other conditions to grow and maintain cell cultures, and 37 degrees is typically chosen because it approximates the normal body temperature of humans. And serial passage, referring to here, this passage right here, is the process of growing bacteria or a quote virus in iterations by growing in one environment, then removing a portion of the population and putting it into a new environment. And this phrase 1:3 diluted vial stock to new cells means one part of the original sample was mixed with 3 parts of the new culture medium. Yeah, so basically they were diluting the environment. So they would dilute these apical samples, passaged or grown in another environment that is at diluted at this ratio 1:3. So 3 parts of the new culture medium versus 1 part of the original. All right, continuing further in the paper. So the cells were monitored daily with light microscopy for cytopathic effects and with RT-PCR or the reverse transcription PCR for the presence of viral nucleic acid in the supernatant. After 3 passages, apical samples and human airway epithelial cells were prepared for transmission electron microscopy. All right, so they grow this 3 times. So put this. So you would grow it, then take some out, put it in a new environment, and then grow it, and then take it out, put it in. Do this 3 times total. All right. And then light microscopy or optical microscopy refers to microscopes that use visible light in a system of lenses to magnify images. And cytopathic effect or cytopathogenic effect or CPE refers to structural changes in host cells, quote, caused by viral infection, such as lysis, which is the breaking down of the cell membrane, or such as cell death without lysis due to an inability to replicate. Interesting. So basically cell death or cell breakdown and so on, cell membrane breakdown. And the assumption is it's from a virus. Interesting. All right, so now let's go over a summary of isolating the quote virus. So let's quickly summarize the quote virus isolation methodology used in this paper. So human airway fluid samples were collected from patients and mixed with a chemical preservative or fetal bovine serum, and the samples were spun really fast in a centrifuge to remove cellular debris, and the supernatant was then removed and used to quote infect human airway epithelial cells obtained from lung cancer surgery patients and grown as a cell culture on a plastic surface. And here's the steps in this inoculation. So prior to quote supernatant infection, the top part of the human airway epithelial cells, or the apical surfaces, was washed 3 times with a water-based salt solution, or PBS, phosphate buffered saline. And then 0.15 milliliters of supernatant was used in the quote infection and was placed on the apical surfaces, or the top surfaces of the cell cultures. And then after 2 hours of incubation at 37 degrees, which is again normal human body temperature, with 5% CO2 in an air-liquid interface, quote unbound virus was removed by washing with 0.50 milliliters of PBS for 10 minutes. And this was at 500 microliters. I'm just going to call it, you convert it to 0.5 or half a milliliter. Then, PBS for 10 minutes. Interesting. Then every 48 hours, 0.15 milliliters of PBS. And I like using milliliters, just it's easier to grasp than microliters, for me at least. So 0.5 milliliters of PBS was applied to the apical surfaces of the cell cultures, then, quote, incubated or grown at 37 degrees for 10 minutes, then harvested, i.e., some apical surfaces of the cell cultures were removed from the culture. Then pseudo-stratified musculary or mucociliary epithelium cells were maintained in the above environment. The removed/harvested apical cells were diluted at a ratio of 1:3 to a new cell culture. And the new cell culture was monitored daily with an optical microscope for cell breakdown or cell death, aka cytopathic effects. And the supernatant of the new cell cultures was monitored with RT-PCR, reverse transcription PCR, for the presence of, quote, viral nucleic acid. And then this above procedure right here was done 2 more times, aka 3 total passages, starting from the supernatant at the last step. So you get the supernatant right here of the new cell culture, and then infecting a new set of human airway epithelial cells at the diluted setup here. Actually, no, the diluted is already done this in the step. So then they would take this and infect a new one, and then go back here, then dilute, and then monitor. And then do this, 2 more times total, so that would be 3 total passages. Interesting. So last step, then quote, infecting a new set of human airway epithelial cells. All right, so after these 3 total passages, the apical cells, or the supernatant, it basically means a supernatant right here. You just take the top part, and bottom human airway epithelial cells were prepared for transmission electron microscopy, or TEM. Alright, so now let's go to the next section of this paper. It's still on the paper methods, but it's going to be on the electron microscope preparation. Just basically the electron microscope is going to be done on the supernatant of these cultures, and the overall cells over there, the bottom part. So anyways, now let's take a look at the next part of the paper, which is preparing the samples for electron microscopy. So transmission electron microscopy. A supernatant from human airway epithelial cell cultures that showed cytopathic effects was collected, and then also inactivated with 2% paraformaldehyde for at least 2 hours, and ultracentrifuged to sediment virus particles. All right, interesting. So virus inactivation refers to the process of removing, quote, viruses completely or rendering them, quote, non-infectious. And paraformaldehyde is a chemical preservative used to, quote, fix proteins or other molecules in the sample via cross-linking polymers, thus locking them in place and preventing any further changes. And fixation is the preservation of biological tissues from decay or degradation to make them suitable for subsequent analysis. And a cross-link right here is a bond or a short sequence of bonds that links one polymer chain to another. And the 2% paraformaldehyde indicates the concentration of the solution, so 2 grams of paraformaldehyde for every 100 milliliters of solution. And an ultracentrifuge, right here, because it's ultracentrifuge, basically a very fast centrifuge with speeds up to 100,000 RPM or revolutions per minute. And transmission electron microscopy, TEM, involves firing a beam of electrons through an ultra-thin section, and the resulting interactions with the sample forms an image that can be made visible. can be made visible through various methods, such as a fluorescent screen, and electron microscopes provide far greater zoom than optical microscopes, but the samples used are of dead material because of the high energy electron beam, and typically stained with heavy metals. And heavy metals have relatively high densities or atomic masses, and this fluorescence right here, fluorescence is the emission of light by a substance that has absorbed electromagnetic radiation. And electrons and electromagnetic radiation, EMR, are not properly defined by mainstream science, according to my opinion. But a good way to view them is as interactions with an all-pervasive medium, the ether or the aether. Electrons can be viewed as vibrations in the ether, and EMR, electromagnetic radiation, as the resulting waves in the ether. So you could think of electrons as vibrations. They have an up and down like this, just like a puddle, and then the radiation is the resulting waves that radiate from that vibration source in the ether. And some good resources on this are Einol, Dino, and Fractal Woman YouTube channels right here. I put the links on there, so check those out. All right, then going further, so here's an image of this transmission electron microscopy. So transmission electron microscope operating principle, basically you would fire a beam of electrons, and then focus it down to the sample, and then the interactions through it, you would focus it back onto this right here, to this fluorescent imaging right here, and you're going to see some dots in there, and this would represent these different grooves and so on of the sample. And then this is a fluorescent plate right there. So the beam is then magnified by other lenses and detected, for example, using fluorescence right here, fluorescence screen. Fluorescence just emits light by a substance that has absorbed electromagnetic radiation. So it absorbs it unevenly, then it's going to reflect, and then it's going to reflect visible light out, or emit visible light out. Then you can see the differences in the contrast and so on. That's very interesting. All right, continuing further in the paper, the enriched supernatant was negatively stained on film-coated grids for examination. So enriched supernatant refers to the portion of the centrifuge supernatant that contains the sedimented virus particles, and the term enriched refers to the concentration of the solute, in this case the virus particles, has increased. So yes, it's enriched right there. So you would use ultracentrifuge. Yeah, so you would take that supernatant, and then you would ultracentrifuge it, so then you would basically get sediment of the supernatant, or get supernatant of the supernatant, and then the sedimented particles are assumed to be virus, and that's called enriched supernatant. Right there, because you're ultracentrifuging it, the already centrifuged supernatant. All right, and now this was negatively stained. So a negative stain involves staining the background while the sample is not stained. In contrast, positive staining involves staining the actual sample. And staining is a technique to increase contrast in samples. In electron microscopy, electron-dense compounds of heavy metals are typically used, and contrast is the degree of differences in color that makes an object visibly distinguishable. And then these film-coated grids are a type of electron microscopy sample grid that has a thin layer coated onto its surface to provide a hydrophilic surface for the sample to adhere to, while the grids themselves are typically made of a thin metal, a metal mesh such as copper or nickel. And hydrophilic, Greek for water-loving, refers to molecules that are attracted to water and tend to be dissolved by water. All right, so yeah, this one just puts a hydrophilic surface on it, and I believe some of them put hydrophobic, water-hating, but yeah, some do this water-loving one as well. All right, so over here is a FarmVar polymer, this type of polymer over there that's coated on this grid. It also has a carbon-coated grid. This is just one type of it right there for these film-coated grids. All right, let's continue further. So continuing further in the paper, human airway epithelial cells showing cytopathic effects were collected and fixed with 2% paraformaldehyde, dash 2.5% glutaraldehyde, and were then fixed with 1% osmium tetroxide, and then dehydrated with grade ethanol embedded with PON812 resin. All right, so there's a lot of technical terms right here and procedures. So glutaraldehyde is another cross-linking chemical compound, and osmium tetroxide is a heavy metal chemical compound used for fixation and or staining. And ethanol is a type of alcohol. And grade ethanol refers to a highly purified ethanol free of impurities that could affect the experimental results. And ethanol is a dehydrating agent that is able to penetrate cell membranes and replace water molecules within the cells with itself and so on. And then this A right here. Actually, I'm not sure why it says AI there. Just put delete this. So PON A12 resin is an industrial resin, and then resin is a solid or highly viscous substance that is typically convertible into polymers. And viscosity is a measure of a fluid's resistance to deformation. For example, syrup has a higher viscosity than water. And embedding samples in a solid support material such as the resin is to provide structural support for the sample during further processing and imaging. Very interesting stuff here. So yes, there's just a lot of technical terms on this. So basically they would take these human airway epithelial cell cultures, collect it, and then you would put these chemical preservatives on them, and then add this heavy metal preservative, then dehydrate it, put alcohol on it, and then embed it in a resin or a gel-like substance and so on. All right, very interesting. So continuing further in the paper, sections 80 nanometers were in thickness were cut from the resin block and stained with uranyl acetate and lead citrate or citrate separately. The negative stain grids and ultrathin sections were observed under transmission electron microscopy. And note that 1 nanometer equals 10 to the negative 9 meters, or 0.001 micrometers. Yeah, so basically ultra thin. So uranyl acetate is a heavy metal stain derived from uranium. And lead citrate is a heavy metal stain derived from lead. Very interesting. All right, so now let's take a look at the summary of electron microscope preparation. So let's quickly summarize the TEM sample preparation methodology in this paper. So supernatant from the quote infected human airway cell cultures that showed cytopathic effects, aka cell membrane breakdown or death or cell death, was collected. The supernatant was quote inactivated with the chemical preservative paraformaldehyde that quote fixes or locks biomolecules in place by quote cross-linking or attaching to polymers. After 2+ hours of inactivation, the supernatant was ultracentrifuged, which involves spinning at really fast speeds of about 100,000 RPM, and this is to sediment the quote virus particles. The portion of the supernatant that contains the sedimented quote virus particles, hence called enriched supernatant, was placed on thin film-coated metal grids that were stained with heavy metals, and then i.e. referred to as negative staining. Human airway cells that showed cytopathic effects were collected and fixed with more preservatives. So this was a supernatant, now it's the actual cells themselves. Yeah, human airway cells that showed cytopathic effects were collected and fixed with more chemical preservatives, fixed with a heavy metal preservative, dehydrated with alcohol, and embedded in a resin resin block. Put a block right here. Resin block to provide structural support. And ultra-thin sections of 80 nanometers, again, extremely thin, were cut from the resin block and stained with 2 heavy metal stains in separate steps. So they would stain one and then stain the other one right after. So if we go right here, stained with uranyl acetate and then stained with lead citrate separately. All right, the centrifuge supernatant on the negative stained grids and the human airway cells on the ultra-thin resin block sections were observed under electron microscopy. All right, very, very interesting stuff here. So now the next part of the paper's methods was on sequencing the quote virus, and then we'll show you the images later and the results and so on. So far, none of the steps covered even remotely show any common sense demonstrations right here of quote isolation, let alone what the paper is trying to achieve. Let's now take a look at the next section of the paper, which is on sequencing the quote virus from the cell cultures. So note that I will not go over too many of the definitions of the specific industry methods for our DNA sequencing mentioned in the paper, because again, I'm more focused on the physical isolation and so on. All right, so RNA extracted from bronchoalveolar lavage fluid and culture supernatants was used as a template to clone and sequence the genome. And we used a combination of Illumina sequencing and nanopore sequencing to characterize the virus genome. And yeah, basically, so it takes the RNA from the airway fluid samples and the cultural supernatants, and then somehow assumes it's a template, or it's used as a template, and then it clones a million times, and then somehow you sequence the genome using these two technologies. And Illumina sequencing involves generating millions of short sequence reads, about 150 to 300 base pairs in length, all in parallel, so doing it all at once. And nanopore sequencing is a new technology that can generate very long continuous sequence reads, several thousand bases in length. And the short reads in Illumina sequencing helped correct errors in the long continuous reads in nanopore sequencing. Yeah, so use these in conjunction. This one is short reads, this one's long ones but has errors. And then you could correct them using these shorter reads. So continuing further in the paper, so sequence reads were assembled into contig maps, which are a set of overlapping DNA segments, with the use of CLC genomic software version 4.6.1 and then CLC bio. And note that a contig, from the word contiguous, is a continuous sequence of DNA that has been assembled from a set of overlapping DNA fragments called reads. And the word contiguous means objects are physically close together, as opposed to continuous means a smooth transition and so on. And a contig map is a graphical representation of the physical organization of contigs in a genome or chromosome. For example, here's a good video tutorial. Here's for example, if you have this chromosome right there, then if you get all the sequences in it, you're going to have a lot of regions overlapping when you're trying to obtain each of these shorter reads. And then when you form a continuous one, that's the contig, which you would have these overlapping ones. So you would have this overlapping over here and so on. I'll just change this color to red. So you have this overlapping, and then you would form a single continuous one. Yeah, like that. And then let's get back to the black pen. All right, so we have that. And continuing further, so yeah, video tutorial, contigs are continuous sequences formed from overlapping sequences. All right, so continuing further in the paper, so specific primers were subsequently designed for PCR, and 5' or 3' race or rapid amplification of cDNA or complementary DNA ends was used to fill genome gaps from conventional Sanger sequencing, and these PCR products were purified from gels and sequenced with a big dye Terminator V 3.1 cycle sequencing kit and a 3130XL genetic analyzer in accordance with the manufacturer's instructions. Interesting. Let's just break this down here. So specific primers were subsequently designed for PCR. So a primer, again, as I went over it earlier, is a short single-stranded nucleic acid used by all living organisms in the initiation of DNA synthesis. And the 5' or 3' right here, this race right here, refer to orientation of a single strand of nucleic acid, and I have this over here. I just showed this illustration. So a furanose or sugar ring molecule with carbon atoms labeled using standard notation. The 5' is upstream, so you go from here 1, 2, 3, 4, 5, and the 3 is downstream. And DNA and RNA are synthesized in the 3, in the 5 to 3 direction. So that's how they're synthesized, starting from here, going downwards, like that. So hence, it's important to know the orientation. And going further here, so complementary DNA or cDNA is DNA synthesized from a single-stranded RNA template by the RT or reverse transcriptase enzyme. In Sanger sequencing or chain termination sequencing involves replicating DNA using modified nucleotides or ddNDPs to terminate the DNA, cDNA, in this case, chain elongation, and allows for generating a series of DNA fragments that differ in length by a single nucleotide. Interesting. So basically, you would replicate, but one nucleotide long at a time, and by these special ones that terminate after you do that one, which is quite interesting. And also, so this primer part right here, here's another illustration of primers. So schematic, the DNA replication fork. So you have RNA primers labeled at the top. So here's RNA primer, and there's a video animation of showing how this works. And the fork splits the DNA double strand. So this fork splits these into 2, like that, double strands. And then the leading strand replicates continuously. So then this replicates continuously, and then you're gonna replicate this, matches up with this. And the leading strand replicates continuously, while the lagging strand replicates discontinuously. This one here adds as one at a time, like that, or it puts this primer like this. Yeah, so this is they're both, this is lagging one, this is the leading strand, and this one adds a primer, and then synthesize adds a primer. I believe that's how it works. But anyways, this one is more discontinuous. This one is continuous setup. All right, let's continue further. So continuing further in the paper, multiple sequence alignment for the 2019 nCoV and reference sequences was performed with the use of muscle. Phylogenetic analysis of the complete genomes was performed with RAXML, and then bracket 13, with 1,000 bootstrap replicates and a general time reversible model used as a nucleotide substitution model. All right, let's make sense of all this. So multiple sequence alignment is a process of aligning the genomic sequences to identify similarities or differences. And then this phylogenetic analysis or just phylogenetics, is the study of the evolutionary history and relationships among or within groups of organisms. And then bootstrap replicates are a statistical technique to estimate the probability of a phylogenetic tree by generating multiple random samples of the original data set. And substitution model is a model describing the changes in DNA sequences over, quote, evolutionary time. And a time reversible model does not care which sequence is the ancestor and which is the descendant. Anyways, that's just that phylogenetic analysis of these genomes and so on. And let's go over a summary of sequencing the, quote, virus. So here's a quick summary of the, quote, virus sequencing procedure in this paper. So RNA from patient airway fluid plus cell culture supernatants were extracted and assumed to be from a, quote, virus. The genome of this RNA assumed, quote, virus was produced using a combination of Illumina sequencing, which produces millions of short sequences, and nanopore sequences, which produces continuous long sequences. And the short reads are used to correct errors in the long reads. And primers, or short nucleic acids that initiate DNA synthesis, were designed for PCR tests. And then DNA fragments from PCR were then sequenced. And then going further, the, quote, virus sequences were compared with reference sequences, and lastly, statistical analysis was performed on the likely, quote, evolutionary history of the, quote, virus genome. And now the question still remains: where is this, quote, virus? How do they know there is a, quote, virus? Why is the paper throwing the word quote, virus around so casually? Hashtag questions. And now the next segment here, I'm going to go off a tangent and basically go over here. So RNA is assumed to be from a, quote, virus. So after spending hours combing through the paper with a fine-tooth comb, trying to comprehend where or what this, quote, virus is, I finally realized that the authors were assuming that RNA detected in the samples were from a, quote, virus. Here I expand further on this assumption. So recall the earlier statement by the paper. So RNA extracted from bronchoalveolar lavage fluid and culture supernatants was used as a template to clone and sequence the genome, and we used a combination of Illumina sequencing and nanopore sequencing to characterize the virus genome. And this was the beginning of this section right here. So RNA extracted of that, and that's this sequencing the virus section. So, and I asked ChatGPT, what does it mean by this? So, so let's ask hashtag ChatGPT to explain what this means exactly. And which is pretty interesting. You could just go and ask it a question. It will try to explain what it is in very direct manner. So MES, I asked it, what does this mean? There's the RNA extracted from bronchoalveolar lavage fluid and culture supernatants was used as a template to clone and sequence the genome, and we used a combination of Illumina sequencing and nanopore sequencing to characterize the virus genome. And ChatGPT states this sentence describes a molecular biology experiment that involved isolating RNA from two sources, bronchoalveolar lavage fluid and culture supernatants, and using it as a template to clone and sequence the genome of virus. And it keeps going on and on, but I just wanted to just skip that. I just wanted to ask it about this RNA. So how do you know the RNA is from a virus? And then right here I forgot to write ChatGPT. So ChatGPT, in the sentence you provided, the assumption is that the RNA extracted from bronchoalveolar lavage fluid and culture supernatants contains viral RNA. This assumption is based on prior knowledge or suspicion that the samples contain a viral infection, or that researchers were specifically looking for viral RNA. So in general, the presence of viral RNA in a biological sample can be confirmed through several methods, including PCR-based assays, These assays use specific primers that are designed to amplify regions of viral RNA or DNA, allowing for the detection of viral genetic material in the sample. And then sequencing, once viral RNA has been detected and amplified, the sequence of the RNA can be determined and compared to known viral sequences in databases, allowing for the identification of the specific virus. In immunological assays, these assays use antibodies that are specific to viral proteins or antigens, allowing for the detection and identification of the virus in this sample. And then it says, in the context of the sentence you provided, it is likely that the researchers used one or more of these methods to compare or to confirm the presence of viral RNA in the bronchoalveolar lavage fluid and culture supernatants before proceeding with the cloning and sequencing of the virus genome. All right, very, very interesting. So yes, there is they just assume it's they assume it's viral RNA based on several methods. One of them is checking for other known viral RNAs using PCR and so on. So a summary of RNA virus assumption. Let's just go over a quick summary of this. So let's quickly summarize the ChatGPT explanations for the assumption of RNA being from viral origin, or quote viral origin. So RNA obtained from patient samples or cell cultures may be assumed to be from a virus if observations that the sample contain a quote viral infection, such as from observing with cytopathic effects, and viral RNA specifically being searched for. So if you're searching for it and you find these sequences, you're assuming it's a virus. Several methods can be used to confirm the presence of quote viral RNA. So PCR amplification of specific regions of previously determined viral RNA or DNA. And then sequenced viral RNA can be compared with known quote viral sequences in databases. And then use antibodies, quote, specific to viral proteins or antigens to identify the quote virus in the sample. So this indirect method. So there's no actual isolation and say, hey, there it is. But instead here's a sequence that we have a database that says it's a virus. So these are some interesting concepts which will need to be explored in more detail in a future video. Nonetheless, it still remains, put this here. Nonetheless, it still remains highly questionable that a bunch of sequences of nucleotides, aka RNA, are somehow assumed to be, quote, disease-causing particles that can infect and replicate through a host and spread to other individuals. And now let's go over the next section. It's on the results. So let's go over the results of the paper. And the first section of the results is discussing the patients. So the first part of the results went over the patients used in the paper/research. And let's take a look at this. So patients, 3 adult patients were presented with severe pneumonia and were admitted to a hospital in Wuhan on December 27, 2019. Patient 1 was a 49-year-old woman, patient 2 was a 61-year-old man, and patient 3 was a 32-year-old man. Clinical profiles were available for patients 1 and 2, and a clinical profile is a detailed summary of a patient's medical history. So that's what they were available for patients 1 and 2. Patient 1 reported having no underlying chronic medical conditions, but reported fever with temperature of 37 degrees to 38 degrees, and cough with chest discomfort on December 23rd, 2019. 4 days after the onset of illness, her cough and chest discomfort worsened, but the fever was reduced, and a diagnosis of pneumonia was based on computed tomographic or CT scan. Her occupation was retailer in the seafood wholesale market. And let's go over tomography is imaging by sections using a kind of penetrating wave. And a computed tomography CT scan uses X-rays to penetrate through the body and measure the loss of intensity through the body. And radiography in general is just a general imaging technique using X-rays, gamma rays, or similar radiation to view the internal form of an object. And here is figure 1 right here. Or actually, I got rid of that figure 1. That was just from Wikipedia there. So basic principle of tomography, superposition free tomographic cross-sections S1 and S2 compared with the not tomographic projection P. So if you have this, these free ones, basically you take a slice from here, and then you'll have an image that shows a slice in this exact region, and then there's another one over here. So that's a tomographic as S1 and S2. But then image P is just looking at it from this way. It's just a projection of all these onto here, which is not a tomographic one because you don't know basically doesn't show. It doesn't show detail in terms of the thickness through it, because this is all just lumping everything together in a 2D image. Versus this one here, if you had a whole bunch of these, you can get a 3D image like this, and so on. But yeah, so you get a slice through that, and you'll see which items are seen in it and are seen on the same plane, and which are not. Very interesting stuff. And going further, here is a CT scanner. Yeah, so you lie down in there and scan through this whole thing. Fascinating stuff there. Anyways, continuing further into the paper. So patient 2 initially reported fever and cough on December 20th, 2019, and respiratory distress developed 7 days after the onset of illness and worsened over the next 2 days. And then see chest radiographs, figure 1, at which time mechanical ventilation was started. He had been a frequent visitor to the seafood wholesale market. And patients 1 and 3 recovered and were discharged from the hospital on January 16, 2020. And patient 2 died on January 9, 2020. And no biopsy specimens were obtained. I believe this is for all of the patients. So respiratory distress is the condition of having trouble breathing. And mechanical ventilation, right here. So this patient 2 had mechanical ventilation. Is the use of a ventilator to provide artificial ventilation, which is the moving of air into and out of the lungs. And discharge right here refers to releasing a patient from a healthcare facility after receiving medical treatment. And a biopsy is the removing of a sample of tissue from a body for examination to determine the presence or extent of a disease. And here is a ventilator right here, and then you just connect it to the person's body and have it breathe for them mechanically. It's very, very interesting. Continuing further in the paper, and we have these chest radiographs right here. That's figure 1. And again, this is for patient 2. And then let's have a look at these radiographs and the information here. So showing our chest radiographs from patient 2 on days 8 and 11, after the onset of illness, and the trachea was intubated, or basically a tube shoved down the throat, and mechanical ventilation instituted in the period between the acquisition of the 2 images. So, for example, here's the first one after 8 days after the illness, and then they got mechanical ventilation, and this is after 11 days of the mechanical breathing. And so you have a bunch of very interesting images there. And then it states here that bilateral fluffy opacities were present in both images, but are increased in density, profusion, and confluence in the second image. And these changes are most marked in the lower lung field, changes consistent with accumulation of pleural, let's try that again, a pleural liquid, are also visible in the second image. So let's, basically what's saying is that it's basically have some fuzzy areas right here and becomes more fuzzy here in the lower part on here. So you can see through it here. Notice after 11 days now it's becoming even more opaque. So the trachea or windpipe is a tube that connects the larynx to the lungs, and intubation is the medical procedure involving the insertion of a tube into the body. And then these bilateral fluffy opacities refer to radiological findings on chest images involving cloudy or hazy areas on both sides of the lungs, indicating various medical conditions. And opacity is the measure of impenetrability of electromagnetic radiation or other kinds of radiation. And over here has increased profusion on the bottom image. Profusion refers to the extent or degree of something, so it's increased more of these bilateral fluffy opacities with those hazy, cloudy areas. And confluence refers to the merging of multiple structure, in this case, fluffy opacity. So you have multiple of these all merging into one region there, or just making all merging together into this image there. You see them all together. And the next one is right here, the pleurae, or plural for this plural. I believe that's plural for it. But anyways, the pleurae are two opposing layers on overlying the lungs and inside the chest wall. And pleural liquid is the fluid inside the space between the pleurae and acts as a lubricant. So if you have the lungs here, and then you have the chest wall around it, and now on this layer there, you're going to have this double layer there. So there's the pleural cavities in between. There's a double layer there, and there's fluid inside this that acts as a lubricant. There's an intercostal muscle. That's these parts right there on the chest wall. It's very, very interesting. Yeah, so this shows that there's accumulation of that pleural liquid inside, in between the lungs and the chest wall, which is supposedly that is what's occurring and making it more opaque from the CT scan. So very, very interesting. All right, continuing further. Yeah, now we're gonna take a look at a summary of patients' results. So here's a quick summary of the patients' results. 3 adult patients with severe pneumonia were admitted in a Wuhan hospital on December 27th, 2019. And patient 1 was a 49-year-old woman. Medical history was available and had no underlying chronic medical conditions. And then had a fever. She had a fever and cough with chest discomfort on December 23rd, 2019. 4 days after onset of illness, her cough and chest discomfort worsened, but fever was reduced. And she was diagnosed with pneumonia based on a CT scan. So she worked as a retailer in a seafood market and recovered and left the hospital on January 16, 2020. Patient 2 was a 61-year-old man, had medical history was available as well, and had a fever and cough on December 20th, 2019. Had trouble breathing 7 days 7 days after illness started. The illness worsened over the next 2 days, which time was placed on, or I'll just fix that up, at which time a ventilator was used to artificially push air into and out of his lungs. And he died on January 9th, 2020. And patient 3 was a 32-year-old man and recovered and left the hospital on January 16th, 2020. It doesn't show much info on this. And no biopsy or tissue samples were obtained from any of the patients. So here's a note here. So 3 patients, a small sample size, again, just 3. No samples taken, and still no, quote, virus proved anywhere. So it's very interesting. All right, and now let's go to the next section of the paper results. This is on detection and isolation of a new, quote, virus. So now let's take a look at the next part of the paper results, this time on supposedly, quote, detecting and isolating a new virus. And here it is, the detection and isolation of a novel coronavirus, and these are the results of these detections and isolations, supposedly. So 3 bronchoalveolar lavage samples were collected from Wuhan Jingtan Hospital on December 30th, 2019, and no specific pathogens, including this is a human coronavirus 229E, HCOV, human coronavirus NL63, then this 0C43, and this other one, HKU1, were detected in clinical specimens from these patients by the RespiFinder Smart22 kit. And then RNA extracted from bronchoalveolar lavage fluid from the patients was used as a template to clone and sequence a genome using a combination of Illumina sequencing and nanopore sequencing, and more than 20,000 viral reads from individual specimens were obtained, and most contigs matched to the genome from lineage B of the genus Beta coronavirus, showing more than 85% identity with a bat SARS-like COV, and its name is here bat SL, and then COV ZC45, and then MG772933.1. Genome published previously. So yes, they basically found some pneumonia patients, took some airway fluid samples, and then used some genetic testing kits to detect if there's these previous supposed coronavirus, human coronaviruses. I believe these are the common flu ones, or the common cold symptoms, and so on. But then no specific pathogens were found, supposedly, and then instead they took RNA from that fluid and then used it as a template to clone and sequence a genome using these 2 sequencing technologies. And they got 20,000 reads, and then most of the contigs or the continuous sequences from those. So you have all these, all of the sequences, and then you have then you'll make a continuous sequence from this right here. Then all these overlapping ones, you'll make it, and and then you form that into one giant sequence. It's a contig, or that's contiguous. So match the genome from lineage B, and that shows 85% identity with this bat SARS-like COV. All right, and again here these types of coronaviruses are types of human coronaviruses, and going of known human coronaviruses. And also this right here, this RNA template to clone, uses a template to clone, refers to creating DNA from a specific RNA sequence and then amplifying the DNA to make amplifying DNA to make many copies. Yeah, so you would use the RNA as a template, produce the complementary DNA, and then amplify DNA to make many copies. And then viral reads, the 20,000 viral reads, are short DNA or RNA sequences generated after breaking down, quote, viral genetic material into small fragments and then using high throughput sequencing technology to sequence the fragment. So yes, a whole lot of sequencing going on. All right, continuing further into the paper. So positive results were also obtained with use of a real-time reverse transcription PCR assay or RT-PCR assay for RNA targeting to a consensus RD-RP region of pan beta coronavirus beta CoV. Although the cycle threshold value was higher than 34 for detected samples, and assuming these are high threshold values there. And virus isolation from the clinical specimens was performed with human airway epithelial cells and Vero E6 and this Ha7 cell lines. So these supposed isolation techniques were done not just on the human airway epithelial, but also on these Vero and these Ha7 cell lines. And the isolated virus was named 2019 nCoV. All right, and now this PCR cycle threshold. So this one here, for this test, they used a relatively high number, even as stated from them right here, although the cycle threshold value was higher than 34 for detected samples. So the PCR cycle threshold, or CT value for short, is a specified maximum number of PCR cycles allowed for a positive reading to be accepted when detecting a specific DNA or RNA sequence, and it can also be used to quantify the amount of DNA or RNA in a sample. And here is a chart of that. So here's the source from this random website. So illustration of cycle threshold CT for, or C and then lower t like that. Yes, capital C, lower t, for specifying a sample as positive or negative for a targeted sequence. So in the PCR one, if you're amplifying, and then each cycle you're going to be amplifying a lot of the DNA that you're replicating. So you're going to replicate, it's going to get a whole bunch, and then after a set amount of cycles, the CT value, and also there's a fluorescence threshold right here. So the way it works is once you amplify enough, and then if it gives off a specific fluorescent radiation emission, and then it'll be considered as positive. But then the number of cycles to get to that point, if it's above the CT value, then you're not going to have a positive sample. Yeah, for example, here, so the CT is right here, and then this sample right here, it was able to cross this threshold, or this fluorescent threshold, before or right at the CT maximum cycle threshold, and then now this is considered positive. But then these other ones, this other one that you're amplifying a whole bunch of times, but then it never reaches this threshold, is a negative sample. And this other one that goes above this, but it took you way too many cycles. And this one is 35.19 PCR cycles. So there's a fraction there. So this is still considered a negative sample. So this one you only need to amplify a tiny bit. So this just means there's more of that sample inside this one to get this positive reading. Like that. So, and interestingly, this example had 35.9 as a negative sample. This one is a 34 still. So then I would assume it's going to be on the high end as well. All right, going further. So right here, so Vero E6. So Vero E6, the quote virus isolation was performed on the human airway epithelial and Vero E6 and Ha7 cell lines. So Vero E6 are a type of cell line derived from the kidney cells of an African green monkey in 1968 as the Vero 7-6 cell line, and then cloned in 1979 to form the Vero E6 cell line. And these cell lines are commonly used in quote virus isolation because they are highly permissive to quote viral infection. And a cell line is a population of cells that are derived from a single cell and can be maintained and grown indefinitely in culture. So it's a population derived from a single one, and you can keep maintaining, growing them, and so on. And Ha7 is a cell line derived from a liver cancer. From the liver cancer, or fix that up, from liver cancer cells of a 57-year-old Japanese male in 1982. Alright, that's interesting. And then cloning cell lines is the process of creating genetically identical cells from a single parent. Alright, it's very, very interesting. So basically, they would do this quote, virus isolation experiments or procedures on the human airway epithelial cells of the patients. These are of the cultures, as well as these green monkey cells, or these cell lines are just these culture cells, from kidney cells of an African monkey, and then that was cloned and kept going for many years. And also this Ha7, this is a liver cancer patient, a Japanese 57-year-old Japanese male with liver cancer. And then he took cells from his liver cancer, and they grew that in a culture, and you get a cell line of those identical cells. Interesting. So, continuing further in the paper, to determine whether virus particles could be visualized in this 2019 nCoV infected airway epithelial cells, mock infected and 2019 nCoV infected human airway epithelial cultures were examined with light microscopy daily and with transmission electron microscopy 6 days after inoculation. So this mock infected, they didn't actually explain how they did that. They just stated that. So cytopathic effects were observed 96 hours or 4 days after inoculation on surface layers of human airway epithelial cells. And there's a lack of cilium beading was seen with light microscopy. Yeah, I believe they just spelled this wrong. There should be an S somewhere there, but I just copied it from the paper. So, seen with light microscopy in the center of the focus, figure 2, and no specific cytopathic effects were observed in the Pharaoh E cell and Ha7 cell lines until 7 days after inoculation. So here's figure 2, cytopathic effects in human airway epithelial cells after inoculation with 2019 nCoV. So this one's mock infected. They don't explain how. They just do that, and then there's just a bunch of uniform dots there. And now the center of this focus, this is the human airway epithelial cytopathic effects, and it just has this light contrast area that's supposedly the cilium beading. And mock infected is a control group in which the same procedure is done but without the actual quote virus. Well, that's the definition, but I don't know what they actually did. And then cilium beading refers to the coordinated movement of cilia, or plural of cilium. And remember, these are the ones that move the mucus and particles out of the respiratory tract. And then this one is they're not moving in coordinated form anymore. But again, this is just a blurry image of something. So continuing further in the paper, electron micrographs of negative stain 2019 nCoV particles were generally spherical with some pleomorphism, so it's having different shapes. Figure 3. So diameter varied from about 60 to 140 nanometers, and virus particles had quite distinctive spikes, about 9 to 12 nanometers, and gave virons the appearance of a solar corona. so these apparently this is the virus of virons, and then there's a solar corona around somehow. All right. Extracellular free virus particles and inclusion bodies filled with virus particles and membrane-bound vesicles in cytoplasm were found in the human airway epithelial ultrathin sections. This observed morphology is consistent with the Coronaviridae family. Alright, so now we have this figure right here, and this is the main quote evidence, the physical evidence besides the sequencing that they're showing that there's a supposed virus. So, visualization of this 2019 nCoV with transmission electron microscopy. So you have negative stained 2019 nCoV particles. This is from the supernatant, is in panel A. Yeah, are shown in panel A. So panel A is a supernatant, and you have these right there. Yeah, those are the supposed virons and the supposed solar corona around it. And you also have on the part B, this is the ultra thin sections of that in that resin block that were cut. And 2019 nCoV particles in the human airway epithelial cell ultrathin sections are shown in panel B. Arrowheads indicate extracellular virus. So those are the arrowheads. No, the arrowheads is right here. So these ones are assuming that these are got out of the cells. And this is another one arrow. Supposedly these little dots are viruses. All right. And what else? Arrows indicate inclusion bodies formed by virus components. So there's inclusion bodies or supposed virus factories inside the cells that somehow produce these supposed viruses. And going further, and triangles indicate cilia. So these are the cilium beating here. All right, this is black and white. Remember, these are all dead images and then stained as well. All right. All right, continuing further. So pleomorphism, it's over here. So some of these show pleomorphism, just showing different shapes. Refer to the variability in the size and shape of cells or their nuclei. And then this solar corona, so the solar appearance of a solar corona, or solar or stellar corona refers to the outermost layer of a star's atmosphere. And during a total solar eclipse, it can be seen by the naked eye. And a total solar eclipse occurs when the moon completely blocks the sun from being seen from a point on the Earth. And extracellular free virus particles are quote viruses found outside of and released by quote infected cells. And these supposedly can be spread through coughing, talking, sneezing, and inhaled by others to start a new quote infection. All right, virus inclusion bodies or viroplasm are supposedly quote viral factories where the quote virus replicates and assembles. So these inclusion bodies are supposedly where these viruses are being produced, supposedly. All right, membrane-bound vesicles are structures within cells that are enclosed by membrane and have various purposes such as transporting, storing, or breaking down material. And the cytoplasm is all the material within a cell. So membrane-bound vesicle, that was over here. So those ones, the virus particles are in the inclusion bodies in membrane-bound vesicles. And going further, here is a solar corona. Yes, the solar corona there. I didn't put any notes there. So this is the solar corona around the. This is when the moon blocks the sun, and now you have these rays. All of these stuff around it, the corona, around the sun there, that's visible in the naked eye, as stated here. Yeah, so a total solar eclipse occurs when the moon completely blocks the sun from being seen from a point on the Earth. And this is just right to here. Total solar eclipse. Yeah, and here I just wrote that down. So total solar eclipse making the solar corona visible with the naked eye. And here's another illustration of a solar eclipse. So you have the sun here. And again, this is from a point on the Earth. That's how it will be visible. And then you have the shadow of it. The shadow, the inner one, is called the umbra. But then whereas the outer one here, cutting from this angle, you have this penumbra. There's an orbit of the Earth and then orbit of the moon around like that. And you're going to have eclipses and so on. And here is wrote that down. So illustration of a solar eclipse. All right, then now let's continue further and now look at a summary of this quote new virus detection and isolation from the paper results. So here's a summary of the paper results on detecting and isolating this quote new virus. So 3 airway fluid samples were collected from a Wuhan hospital on December 30th, 2019, and no specific quote viruses were detected from these patients or specific known viruses. Were detected from these patients, and an RNA was extracted from the airway fluid samples and assumed to be from a quote virus. The RNA was used as a template to create DNA, which was later amplified to many copies, and then sequencing technology was used to produce a genome. All right, and then 20,000 short RNA sequences were obtained, and long continuous sequences from these short and overlapping sequences, i.e. contigs, were produced and showed 85% similarity with a bat coronavirus. Then PCR tests using a cycle threshold CT value higher than 34 were positive when using a known sequence common among beta coronaviruses. So first of all, they did tests and they showed that no specific known viruses were detected from those patients. And then they showed sequences from beta coronaviruses that were shown inside samples using these PCR tests, and then the overall long continuous contigs, or pretty much genomes, showed 85% similarity with a bat coronavirus. And quote, virus isolation from the clinical sample specimens were performed on human airway epithelial cells, monkey kidney cells, and liver cancer cells from a 57-year-old Japanese man. Cancer. So yeah, monkey cells and also from liver cells from a 57-year-old man. And the quote, isolated virus was named 2019 nCoV, or 2019 novel coronavirus. And the quote, virus was then used to quote, infect human airway epithelial cell cultures with a quote, mock infected culture used as a control, although there's no information shown in the paper about this. They just state it, but they don't actually show anything else. The cultures were examined daily with optical light microscopy, and with TEM, or transmission electron microscopy, 6 days after quote, infection. And cytopathic effects were observed 96 hours, or 4 days after inoculation. And also, I just want to go back here. So the cultures were examined daily with optical light microscopy and with transmission electron microscope, 6 days. I think this means 6 days after infection, or 6 days. So you would basically, so because it's doing it daily, I think there's the word 4, but then if you scroll back up to what it actually said, they never actually said here. So with transmission electron microscope, 6 days after inoculation, but it was searched daily. And I'm just going to go 4 ? I'll just put that in. I think that's what it means. Because the cytopathic effects were observed 96 hours after inoculation, even though this says 6 days after. So obviously it was doing it daily. All right. Yeah, so we corrected that part of the paper. So yeah, cytopathic effects were observed 96 hours or 4 days after inoculation on surface layers of human airway epithelial cells, and with a lack of coordinated cilium movement was observed in some spots. So that's cilium beating. In the monkey kidney and human liver's cancer cell cultures, cytopathic effects were observed 6 days after quote infection, as opposed to 4 days for the human airway samples from the patient. So, and here's the images. There's the mock. There's the human airway epithelial with showing the cytopathic effects. Again, it just shows just a little low contrast area. So figure 2, supposed cytopathic effects, CPE, in human airway cell culture. On the right image with a lack of cilium beating in the center, and there's the mock on the left. For the supernatant samples, electron microscope images of quote virus particles right here were presented, in which the background was stained with heavy metals. The particles varied in size and shape and ranged between 60 to 140 nanometers. Supposedly with spikes 9 to 12 nanometers in size, and supposedly looking like a solar corona. This just looks like just something. For the embedded and heavy metal stained / fixed thin sections of human airway epithelial cell cultures, electron microscope images showed quote viruses. I'll put this right here. Yeah, put quote viruses outside of the cells, as well as within membrane-bound structures inside the cells that supposedly act as quote virus factories in the replication and formation of quote viruses. The observed quote virus particles supposedly belong to the Coronaviridae family. So right, so here's the 2 images. So negative stained supernatant on the left, supposedly showing quote virus particles, and on the right shows the ultrathin embedded sections. Inclusion bodies or quote virus factories within cells. That's the arrows. There's extracellular viruses, arrowheads, and there's cilia, the triangles right here. All right. So yes, so this is the supposed evidence of a new virus that's supposedly infecting the world, and to me, this is not evidence. Alright, and now let's continue further and take a look at the next part of the paper results that's on the genetic characterization of the quote virus. So this section is still in the quote detection and isolation of a novel coronavirus results part of the paper, but it dealt specifically with genetic sequencing, so I'll cover this separately. So going further, so to further characterize the virus, de novo sequences of 2019 nCoV genome from clinical specimens, bronchoalveolar lavage fluid, and human airway epithelial cell virus isolates, presumably referring to the cell culture supernatant, were obtained by Illumina and nanopore sequencing. The novel coronavirus was identified from all 3 patients. But this de novo sequencing, and this de novo sequencing refers to genetic sequencing without using any existing reference or template sequence, and the process involves DNA or RNA molecules being broken into smaller fragments, sequencing those fragments, and then a computer algorithm assembles the fragments into a, quote, complete genome sequence. Alright, so that's basically, when they're speaking about isolating the virus, what they're talking about is some next level genetic sequencing. And then the question is, well, what do they mean by this? So continuing further in the paper. So 2 nearly full-length coronavirus sequences were obtained from bronchoalveolar lavage fluid, and this is called beta-CoV Wuhan, and so on, this identification. And that's this one, and there's the second one right here. And 1 full-length sequence was obtained from a quote, virus isolated from a patient, and this is this one right here. This is the full length, the other one is partial length. But then they state that complete genome sequences should be 2 partial, 1 complete, because it says right here 2 nearly full. Yeah, so we have 2 nearly full and 1 full. And then 1 full length sequence were obtained. Then it says complete genome sequences should be 2 and not complete sequences of the 3 novel coronaviruses were submitted to GISAID, and it's this one right here they sent, 402119, that's this one. And they sent this one to the 20, this one right here, and there's the other one here, 40212. No, actually that's a different ID. So this one right here, let's see where this one is. Yeah, this one here. So 40212. That's 2121. That's this one. There's a 20. That's they don't have that one there. Where's this one? This one right here is this. And going further, yeah, this one right here is the 22. Actually, I'll see that again. So this right here. Oh, I'm going to underline this one. Is this? I just want to be double check, make it clear. And the next one is going to be this right here. Actually, this one right here. I just want to see the numbers. APSL. I'll underline this. Which ones match? And this one matches. And the next one is right here. But that's not matching. This one isn't going to match. Yeah. That's this right here. Alright, so we got these and then a whole bunch of different lingo there. And have an 86.9% nucleotide sequence identity to a previously published bat SARS-like CoV, and that's this coronavirus genome. And the 3 2019 nCoV genomes clustered together with the sars-cov subgenus, which shows the typical beta coronavirus organization, and a 5-untranslated region, UTR, replicates complex ORF1AB, S gene, E gene, M gene, N gene, then R-UTR, that's the untranslated region, and several unidentified non-structural open reading frames. Alright, that's this ORF, open reading frame. So, I'm not going to go too much into what those all mean, but basically I'm just going to summarize that. So first of all, GISAID, or Global Initiative on Sharing All Influenza Data, is a worldwide database for genomic data on influenza, quote, viruses. An influenza, or, quote, the flu, is supposedly an infectious disease caused by, quote, influenza viruses. And the genetic organization mentioned over here, so this Ebola organization, and then I have over here, so the genetic organization mentioned above are the supposed regions of the genome that are either non-coding or code for specific functions of the, quote, virus, such as replication or producing the, quote, spike protein. So this replicates, yeah, that's for replication, and so on. And that's the producing that enzyme, and so on. All right, so now continuing further in the sequencing part. So, although 2019 nCoV is similar to some beta coronaviruses detected in bats, and that's figure 4, I'll go to that a bit, it is distinct from SARS-CoV and MERS-CoV. The three 2019 nCoV coronaviruses from Wuhan, together with two bat-derived SARS-like strains, ZC45 and ZXC21, form a distinct clade. And SARS-CoV strains from humans and genetically similar SARS-like coronaviruses from bats collected from southwestern China formed another clade within the subgenus sarbacovirus. Since the sequence identity in conserved replicase domains ORF1AB is less than 90% between 2019 nCoV and other members of beta coronaviruses, the 2019 nCoV, the likely causative agent of the viral pneumonia in Wuhan, is a novel coronavirus belonging to the sarbacovirus subgenus of Coronaviridae family. Basically, they're saying these supposed conserved domains, or this is the replication domains, part of the gene of many of these coronaviruses. It's less than 90% of between the 2019 nCoV and other members of it. They're going to say that. Yeah, they're going to say that it's less than 90%, so they're going to call it a new virus. So it's so since it's less than the 90% of that conserved region in many of these beta coronaviruses, they're going to call it a new beta coronavirus belonging to this other subgenus and so on. Anyways, going further, so replicase domains ORF1AB are a set of conserved regions in the genome of the coronaviruses that encode for proteins involved in, quote, viral replication. All right, continuing further in the paper, and so here's how the whole genome setup looks like. So this is the beta CoV, this is the 402119 with 29,892 base pairs. Where's that number? Here, this is right here, 402119. So that's the number 402119. And there's the base pairs all there. And there is the phylogenetic tree, and this is the beta one. This is the 2119 in red. So that's the one, and it forms it by itself. So it's different from all these other ones. And yeah, so there's a whole bunch of classification. And there's the MERS is over here. So MERS over there, and these are the SARS-CoV-1. Now these are the beta coronavirus, the new ones. It's going to be somewhere in there. Interesting. So figure 4, schematic of 2019 nCoV and phylogenetic analysis of 2019 nCoV and other beta coronavirus virus genomes. As shown are a schematic of 2019 nCoV, panel A, and full-length, this is panel A, yeah, this is right here, nCoV, and full-length phylogenetic analysis of 2019 nCoV and other beta coronavirus genomes in the orthocoronavirinae subfamily. Yes, very, very fascinating. Yeah, so you have a whole bunch of these other these subtypes. So you have the sarbacovirus, sarbacovirus, and that's the beta one. The new SARS one goes inside there. There's the bat SARS like it, and there's a coronavirus one in there. So interesting. Note clad, or here it says note the clad with the beta CoVs. Is this one right here? The supposed quote novel COVID virus, and you also have underlined bat SARS-like coronaviruses over here. So yes, it's similar to these bat-like coronaviruses. Anyways, going further, so summary of this genetic analysis part, or genetic characterization part. So summary, let's go over summary of the paper results section on genetic characterization of the virus. All right, and here's change the title name. So genetic sequencing of the clinical lung fluid samples. Or just to make it more consistent, I'm just going to call that the airway fluid samples. That's the bronchoalveolar lavage fluid. And the cell culture supernatant obtained the, quote, virus genome. So they did genetic sequencing, and it was that de novo setup. And the, quote, virus genome was identified from all 3 patients. And 2 nearly full-length, quote, virus sequences were obtained from the lung fluid samples. 1 full-length, quote, virus sequence was obtained from the cell culture supernatant. And the 3, quote, virus genome sequences were sent to the GISAID Global Flu Virus Database, and the genome sequences had an 86.9% nucleotide sequence identity to a previously published bat coronavirus genome. the three quote virus genomes form a distinct clade or grouping with less than 90% similarity of genetic sequences, genes that code for proteins involved in quote viral replication. Yes, very interesting. So yeah, so the main replication gene, it has 90% similarity, and then that's why it's going to be calling it a new coronavirus. It's not matching up with the other ones. All right, and now let's take a look at the paper discussions, the last part of the paper, and then I'm going to start summarizing everything. Now let's go over the paper's discussion section to see how they summarize their findings, or the quote findings. So they state that we report a novel COV, the 2019 nCoV, that was identified in hospitalized patients at Wuhan, China, in December 2019 and January 2020. Evidence for the presence of this virus includes identification in bronchoalveolar lavage fluid in 3 patients by whole genome sequencing, direct PCR, and culture. The illness likely to have been caused by this COV was named novel coronavirus infected pneumonia, NCIP, and complete genomes were submitted to GISAID, and phylogenetic analysis revealed that 2019 nCoV falls into the genus beta coronavirus, which includes coronaviruses SARS-CoV, bat SARS-like CoV, and others discovered in humans, bats, and other wild animals. And we report, quote, isolation of the virus and the initial description of its specific cytopathic effects and morphology. And direct PCR right here, because it was detected by direct PCR, refers to using PCR directly on the sample without prior DNA extraction or purification steps. Alright, and now continuing further into the paper, so going here, some molecular techniques have been used successfully to identify infectious agents for many years, and unbiased high-throughput sequencing is a powerful tool for the discovery of pathogens. Next-generation sequencing and bioinformatics are changing the way we can respond to infectious disease outbreaks, improving our understanding of disease occurrence and transmission, and accelerating the identification of pathogens and promoting data sharing. We described in this report the use of molecular techniques and unbiased DNA sequencing to discover a novel beta coronavirus that is likely to have been the cause of severe pneumonia in 3 patients in Wuhan, China. So basically, they look at some patients, test for known sequences, and then they show that okay, it's not there, and then they're gonna test for the sequences that are present, and then be like, all right, we're gonna somehow use a computer algorithm and form a full sequence, say this is a new virus, and assume that's the cause of the pneumonia. All right, going further here, this bioinformatics. I'll fix this up. Yeah, here, just fix that up. So bioinformatics is the field that develops methods and software tools for understanding big sets of biological data. Yeah, that's basically big data for biological systems. And continuing further in the paper, so although establishing human airway epithelial cell cultures is labor intensive, they appear to be, they appear to be a valuable research tool for analysis of human respiratory pathogens. Our study showed that initial propagation of human respiratory secretions onto human airway epithelial cell cultures, followed by transmission electron microscopy and whole genome sequencing of culture supernatant, was successfully used for visualization and detection of new human coronavirus that can possibly elude identification by traditional methods. All right. Visualization just shows some blurry particles. Further development of accurate and rapid methods to identify known respiratory pathogens is still needed, and on the basis of analysis of 3 complete genomes obtained in the study, we designed several specific and sensitive assays targeting the ORF1ab, N, and E regions of the 2019 nCoV genome to detect viral RNA and clinical specimens. And then they would send these sequences and assays to the world, and they would basically go and start testing for these this new virus, quote virus. So the primary sets and standard operating procedures have been shared with the World Health Organization and are intended for surveillance and detection of 2019 nCoV infection globally and in China. And more recent data show 2019 nCoV detection in 830 persons in China. Interesting. So they've already started testing people. Although our study does not follow Koch's postulates, our analysis provides evidence implicating 2019 nCoV in the Wuhan outbreak. And note here, so basically it just says it doesn't follow Koch's postulates. In Koch's postulates are 4 criteria designed to establish a causal link between a microbe and a disease, and are listed below. So the number 1 is a microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. And the microorganism must be isolated from a diseased organism and grown in pure culture containing only a population of that microbe. But for supposed viruses, they need a host. So that's a modification of the Koch's postulates. So the cultured microorganism should cause disease when introduced into a healthy organism. And the microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original specific causative agent. And basically to summarize, basically you gotta find a microbe in abundance in all the organisms suffering from the disease, but not in the healthy organisms. But remember, with viruses and even some bacteria, you can have asystematic carriers. Which you can have that microorganism in healthy organisms, which eludes reasoning. And also then once you isolate it from the disease, it has to be grown in pure culture, and then the cultured organism should cause disease when introduced in a healthy organism, and then you should be able to re-isolate it from that and show that it's the same thing as the original one. That's Koch's postulates in a nutshell. And note that the postulates are not strictly followed anymore, and terms such as, quote, asystematic carriers, which are carrying the microbe but not the disease, and showing that there's a questionable causal link, and, quote, viruses which will require host cells to, quote, grow, are assumed in mainstream science. So you assume a virus existence and you assume asystematic carriers. Yeah, so the asystematic carrier, this is cancels out step 1. And then step 2 is the viruses. So you can have Koch's postulates as originally formatted with viruses. Continuing further in the paper, additional evidence, presumably yet to be done, to confirm the etiological or cause of disease significance of 2019 nCoV in the Wuhan outbreak include identification of a 2019 nCoV antigen in the lung tissue of patients by immunohistochemical analysis, and detection of IgM and IgG antiviral antibodies in the serum samples from a patient at 2 points to demonstrate seroconversion, and animal or particular monkey experiments to provide evidence of pathogenicity. Of critical importance are epidemiological investigations to characterize transmission modes, reproduction interval, and clinical spectrum resulting from infection to inform and refine strategies that can prevent, control, and stop the spread of 2019 nCoV. Very interesting wording, as that's pretty much what's blustered for past few years during the COVID saga. Going further, so this 2019 nCoV antigen, right here, identification of that refers to the proteins or regions of proteins such as the, quote, spike protein, and on the, quote, virus that can serve, or the spike protein, such as the spike protein on the, quote, virus that can serve as antigens and, quote, stimulate the immune system. So those are the antigens, basically specific proteins or regions of proteins on the virus. I'll put this in bracket. Yeah, I'll put this in bracket like that, such as the spike protein. So the spike protein can be viewed as a antigen. And again, the spike protein, whatever, that's supposed to be the part of the solar corona around the virus, but that's questionable in my opinion as well. Going further, so immunohistochemical analysis, or IHC analysis, is a technique used to visualize and detect specific proteins or antigens within tissue sample. Yeah, so this is basically detecting the proteins and antigens and so on. Immunoglobulin M, or IgM, is the largest antibody and first antibody to appear in the response to initial exposure to an antigen. And immunoglobulin G, Ig, capital G, is the most common type of antibody found in blood circulation, and it represents 75% of serum antibodies in humans. And seroconversion is the development of specific antibodies in in the blood, in the blood serum as a result of quote infection or quote immunization, including vaccination. So basically, you have antigen, and then seroconversion is when you build up these antibodies in the blood serum as a result of that antigen, and supposedly that the antibodies inside, they can go and neutralize that specific antigen. Again, that's the mainstream theory. So immunization is the assumed process of strengthening an individual's quote immune system after initial exposure to an quote infectious agent. And pathogenicity is a measure of how effective a pathogen is at causing disease. So yeah, basically they're saying they need monkey animal experiments and basically try to infect them with these supposed viruses, as well as other procedures that likely involve torture. Yeah, because when you read up on how they actually test on animals, it is pretty brutal. Going further, so transmission mode right here too, because they want to determine transmission mode of this supposed virus, is the specific way a disease or pathogen is transmitted to another person or organism. And then reproduction level is the average time it takes for an infected individual to quote transmit the disease to others. And this clinical spectrum is just a full range of signs, symptoms, and outcomes that can occur as a result of a quote disease or condition. All right, let's continue further. So summary of paper discussion of the paper's discussion. So let's go over a summary of the paper's discussion section and let's break it down. So a novel or new CoV coronavirus was identified in hospital patients in December 2019 and January 2020. Evidence for this quote new virus includes identification in lung fluid samples or more accurately airway fluid samples by the following methods: whole genome sequencing, PCR directly on samples, and cell cultures from the samples. Complete genomes were submitted to the GISAID, and the quote new virus falls into the beta coronavirus classification. Modern genetic sequencing and big data analysis technology is quote accelerating the identification of pathogens such as this quote new virus that is quote likely to have been the cause of severe pneumonia in 3 patients in Wuhan, China. Human lung fluid samples, with the assumption that they quote contain a new virus, were used to quote infect human airway epithelial cell cultures. Quote virus particles were assumed to be visualized via an electron microscope, and whole genome sequencing was performed on the cell culture supernatant. From the 3 quote complete genomes, the authors developed techniques to identify this quote new virus by targeting specific sequences from these genomes in the targeting of quote viral RNA in future patients. The sequencing and procedures were shared with the World Health Organization, the WHO, for the surveillance and detection of this quote new virus globally. As of the paper's publishing, 830 people in China tested positive for these targeted sequences. The authors admit their paper does not follow Koch's postulates of a more concrete link of microbe to disease causation. The authors instead claim their study, quote, implicates the, quote, new virus in the Wuhan, quote, outbreak. Alright, so if you scroll back up to see that part, there's the Koch's postulates. So our study does not follow Koch's postulates. Our analysis instead provides evidence implicating 2019 and CoV, alright, so we have that implication. Going further, additional experiments need to be done to confirm the disease-causing claim of this new virus, and these are identify proteins on the quote virus that serve as antigens in the lung tissue of patients, detect corresponding antibodies or proteins in the blood of patients that form to quote identify and neutralize the quote virus antigens. So go look in the blood and look at antigens in the lung tissue. And animal or particularly monkey experiments to demonstrate the virus actually does actually causes disease. And the paper concludes by highlighting the importance of epidemiological investigations to quote investigation to characterize the transmission and clinical symptoms of this quote disease in order to refine strategies to quote stop the spread of this new virus. Interesting. So yeah, I'm just paraphrasing that. And now finally, so we went through the paper, we went through all the summaries of of every section of it. Now I'm going to go over a summary it's my own summary of the entire key parts of the paper. So this video has served as a study of mainstream virology in general for me, and I hope it has for you as well. While the video covers the paper in its entirety, much of it discusses assumptions, theories, and narratives behind the specific experiments actually, beyond the specific experiments actually performed in the paper. Yes, and so now to better grasp the key claims of the paper, I will summarize only the main parts of the paper's most important claim, which is the assumption of a, quote, new disease-causing virus. So ChatGPT, so here's a summary. ChatGPT says the following paper is the main paper on the isolation of the, quote, virus that causes COVID-19. Here's a paper link from the New England Journal of Medicine right there. So a novel coronavirus from patients with pneumonia in China 2019 by Zhu et al. And Latin, the al is Latin for and others. Published on January 24th, 2020. Updated on January 29th, 2020 and appears in the February 2020 issue of the New England Journal of Medicine, NEJM. And so on December 31st, 2019, the China CDC sent a rapid response team to Wuhan to investigate reported clusters of patients with pneumonia or inflammation of the lungs of quote unknown cause and had recently been to or in contact with someone because they were linked to it, that had been to the Huanan seafood market in Wuhan. This paper reports the results of the investigation. The chest radiographs of a patient whom no samples were taken from, no biopsy samples were taken from, with pneumonia and assumed to be from the, quote, new virus is shown below. So these are chest radiographs of it. Yeah, so there's no biopsy or samples taken from them, but there's just some images and assumed that it's from the new virus, and it just shows this cloudy stuff. So figure 1, patient 2 on days 8 and 11 after start of illness, mechanical ventilation or assisted breathing was performed between the time periods of the two images, and the cloudy or hazy areas more present in the bottom image supposedly indicate accumulation of fluid around the lungs. It was becoming more hazy, and again, this period between this image and this image had mechanical ventilation. So that may be a factor too. So for lower respiratory tract, throat / lungs region, samples were collected from the unknown cause pneumonia patients who were in Wuhan on December 21st, 2019 or later and had been present in the market. 7 samples were collected from known cause pneumonia patients from Beijing to be used as control samples. Nucleic acids were extracted from both sets of samples, and PCR so extracted from both of these. So you extract from the patients, from the 4 samples from those patients from Wuhan, and then 7 samples from the Beijing control samples. And they were extracted from them. So PCR DNA replication tests were done on the nucleic acids to test for known sequences from 22, quote, viruses and 4 bacteria, and presumably they weren't found in the Wuhan ones. Yeah, and I just wrote that here. So presumably they weren't found in the Wuhan samples. And then modern whole genome sequencing technology was used to, quote, discover sequences not identifiable by the above PCR methods. And then RT-PCR, reverse transcription PCR, basically goes from RNA conversion to DNA. Then DNA replication assay was used to detect, quote, viral RNA by targeting a specific genetic sequence that is common to many beta coronaviruses. After, quote, unknown sequences after discovering, quote, unknown sequences and, quote, viral RNA, the next step was to, quote, isolate the virus. And then basically had respiratory samples were collected from unknown cause pneumonia patients. And then you had chemical preservatives were added, possibly including fetal bovine serum, to the samples. Samples were centrifuged to remove, quote, cellular debris, and the supernatant was collected. Now you have passage 1. The supernatant was used to, quote, infect human airway epithelial cells obtained from lung cancer surgery patients. And, yes, and then the next step was the airway cells right here. Yeah, these airway cells, which, again, the supernatant from the patient samples were used to infect these human airway epithelial cells grown from these lung cancer surgery patients, and then used to infect them. So the airway cells were initially grown on a plastic surface in an air-liquid interface, bottom in contact with liquid and the top with air, for 4 to 6 weeks. Prior to supernatant infection, the top parts of the airway cell cultures were washed 3 times with a water-based salt solution, this is phosphate buffered saline or PBS, and then 0.15 milliliters of supernatant was placed, aka infected, on the top surface of the cell culture, and of the cancer patient cell culture. The supernatant infected cell culture was then placed in an incubator at 37 degrees Celsius with 5% CO2 in an air-liquid interface. After 2 hours of incubation, quote, unbound virus was removed by washing with 0.5 milliliters of PBS for 10 minutes. Every 48 hours, 0.15 milliliters of PBS was added to the top surface, or the apical surface, or the apical part of the airway cells. And then after 10 minutes of incubation at 37 degrees Celsius, the apical samples, or the top surface of them, were harvested, or i.e. removed. So you remove them after 10 minutes. The removed apical top surface samples were diluted at a ratio of 1:3 to a new cell culture and monitored daily for cytopathic effects, cell breakdown, cell death via an optical light microscope. And then viral nucleic acid in the supernatant was also being tasted via RT-PCR, basically going from RNA to DNA, then replicating. So that's how they were testing. They're monitoring daily for that. And there's the mock, and there's the human airway epithelial cell. So figure 2, mock infected, which the paper doesn't elaborate any further, on the left, and quote, new virus infected human airway epithelial HAE cell cultures on the right. Visualized with an optical light microscope, and then cytopathic effects, CPE, were supposedly observed 4 days after quote infection. And the arrow points to a lack of coordinated movement from cilia, or hair-like structures on the surface of epithelial cells that move mucus and trap particles out of the respiratory tract. So supposedly, that's CPE. Alright, the supernatant in the last step was then removed and used to, quote, infect another airway cell culture, and then repeated again for a total of 3 passages. After these 3 passages, the supernatant from the airway cell cultures that showed these cytopathic effects were then collected. The supernatant was then quote, virus inactivated for at least 2 hours by chemical preservatives, by the chemical preservative paraformaldehyde, which, quote, fixes or locks molecules in place, preventing further changes, aka prevents, quote, virus replication. Then the supernatant was ultracentrifuged to sediment, quote, virus particles. This sediment is called enriched supernatant. The enriched supernatant was then placed on thin grades stained with heavy metals to provide contrast for electron microscope imaging. And similarly, after 3 passages, the airway cell cultures themselves, and not the supernatant, that showed cytopathic effects were collected. The airway cell cultures were fixed with chemical preservatives, paraformaldehyde with the chemical preservatives paraformaldehyde and glutaraldehyde. Then they were fixed, glutaraldehyde. Then they were fixed with the heavy metal preservatives osmium tetraoxide. And they were dehydrated with ethanol alcohol, which penetrates cell membranes and replaces water and replaces water molecules within the cells. It replaces water molecules within the cells. Then they were embedded in the resin PON8112, and then ultra thin 80 nanometer sections were cut from the resin block and stained with the heavy metals uranyl acetate and lead citrate. The negative stain grids containing the supernatant and ultra-thin sections containing the airway cell cultures were observed with an electron microscope. Alright, so you got the supernatant and you got this ultra-thin sections here, the airway epithelial cells that were supposedly infected. And again, remember, these airway cells are from the cancer patients. So electron microscope images, so A, quote, virus particles from the supernatant. And then B, airway cell cultures, and then you have the arrowheads indicate extracellular viruses. That's this right here, extracellular. Then arrows indicate inclusion bodies or, quote, virus factors within cells. It's right here. Then triangles indicate cilia. That is the hair-like structures on the epithelial cells. And the, quote, virus particles termed virion supposedly showed, quote, spikes on the outer surface and supposedly give the appearance of a solar corona. So this supposedly gives the appearance of a solar corona. Hashtag supposedly. Yeah, so these virus particles termed virions supposedly show spikes. They show these little thingies on the outer surfaces, and then supposedly give the appearance of a solar corona. Hashtag supposedly indeed, because this wouldn't pass a video on Math Easy as evidence. Anyways, going further, so RNA was extracted from the patient airway fluid samples and cell culture supernatants. Yeah, so from both of them, from the patient airway fluid samples of the patients and the cell cultures supernatants, this RNA was assumed to be, quote, from a virus. This RNA was used as a template for modern sequencing technology to produce a, quote, virus genome. This novel virus genome was identified in, quote, all 3 patients, and the 3 genome sequences were sent to the GISAID, or the Global Initiative on Sharing All Influenza Data. And then, global quote, flu virus database. Statistical and computational analysis were performed on a likely evolutionary history of the quote, virus genome. And then the 3 genomes have a 86.9 similarity with a previously published, quote, bat coronavirus genome. So there is the 29,892 base pairs of this SARS, of this COVID-19 virus genome, this novel coronavirus. All right, and then this is in red. So the red beta coronavirus are the grouping for the supposed, quote, novel COVID virus. So this right here. So there is the there is the supposed virus right there. The new one, that's the grouping. They somehow got all that in there. All right, finally, let's go to the MES conclusions and try to make sense of all this. So MES conclusions, after sifting through this paper with a fine-tooth comb, the only thing clear is that there's nothing clear about the paper, the methods, or virology in general. A handful of patients were assumed to have a, quote, viral infection. A handful of respiratory samples were obtained and assumed to contain a, quote, virus. Genetic sequencing, aka listing of molecules, and comparing with public database of other genetic sequences was used as a basis for identifying this, quote, new virus. Respiratory samples were heavily processed, centrifuged, mixed with chemical preservatives, grown and regrown in cell cultures derived from cancer patients, stained with heavy metals, washed with saline solutions, embedded in resin, cut in ultra-thin sections, and placed in high-powered electron microscopes. Optical light microscopy showed non-persuasive information, to say the least. So there's nothing there in those images. I'll just write here. Optical light microscopy images. It showed non-persuasive images. Yeah, they just showed that supposed cilium beating. Electron microscope images showed dead particles which are assumed to be, quote, virions. Besides requiring further research into the, quote, genetic sequencing science being performed, there's not much of any significance presented in this paper, and much less what would be expected to declare a, quote, pandemic, lockdown, mask up, and vax up the world. Or rephrase that again, much less what would be expected to declare a, quote, pandemic, and lockdown, mask up, and vax up the world. All right. And interestingly, there are some very peculiar timings of this whole COVID psychological operation, or PSYOP, and PSYOP it most certainly is. So December 1st, 2019, China's new strict vaccine law goes into effect. All right, into effect. And then December 1, 2019, patient 0 started to have symptoms. And then December 31st, 2019, again, just before the new year, China sends a rapid response team to Wuhan. Very interesting. And Wuhan, so the Wuhan right there, Wuhan is a MES anagram. I call it MES because if you move this around, you get HU. Yeah, so the HU, then the HU, and then this one here, flip it, call it an M. W becomes an M, then you have an A, then you have an N, human. So Wuhan, MES anagram, human. So yes, basically a human, imagination right here to create this supposed pandemic. And of course, the paper ends with a now commonplace brainwashing propaganda urging the public and governments to, quote, prevent, control, and stop the spread of 2019 nCoV. Anyway, stay tuned for MESScience 5 and see what I get up to on that video. That's all for today. Hopefully you learned and was able to grasp this paper. It took me a while to grasp it, but yes, it looks like I'll have to do a video on genetic sequencing, and then also get my hands on some genetic sequencing technology and see what we could find. But even then, there's no evidence shown here, at least, of the supposed sequence or virus actually doing anything. actually causing illness and infection and spread and so on. But suddenly this paper, they already have primers and sequences designed for people to test. That's why they said that 830 people already tested positive. And again, with the PCR test, you're just amplifying billions of times whatever sequence you're looking for and the sequence. And the body has a whole bunch of these random sequences. So if you have just one tiny one, you can amplify it and call it test positive. Remember, these go from 2, 4. You go here 2, 4. Yeah, first cycle. This goes 1, then goes 2, then goes to 4, then 8, 16, 32, 64, 128, etc. Anyways, continuing further, I'm just going to scroll all the way back up. And yeah, thanks for watching. And hopefully you'll learn. Let me know what you think. And let me know what you think of this whole COVID situation. And hopefully this video doesn't get demonetized and censored and banned and so on. And also I'll be uploading sections. I'll be splitting this whole video into sections and uploading them. And yeah, just to make it, you make more content for math easy. And also yeah, get more, just get it more out there and easier to digest, because this is a long video. I'm going to upload many shorter ones. And so on. Anyways, thanks for watching, and that's all for today. And again, you can download the notes and view these notes in a Hive blockchain. All the link in the description below. And thanks for watching, and stay tuned for another Math Easy solution.